and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are
here. There we go. Okay. I'll just go ahead and start then. So welcome everybody to day 2 of our Regulatory Education for Industry, or REdI Annual Conference 2026, Innovative Regulatory Strategies to Advance Medical Products. This is the CDER room, so make sure if you are planning to be in a different track, you go ahead and take note now. We're all good. We'll go ahead and keep going. So to those who are online right now, good morning, good afternoon, or good night, depending on wherever you may be. We do appreciate your commitment on attending both days of this event, if you've done so, and also overcoming your busy schedules to join us today. For those who are here with us in person, you can note the on-site Wi-Fi here on the screen. There's no password to join. All you need to do is check the acknowledgement in order to join. My name is Corey Adair, and I am with the Small Business and Industry Assistance, or SBI program, within the Division of Drug Information within the Center for Drug Evaluation and Research, or CDER. It's a great pleasure, along with my SBIA team and our other colleagues within FDA, to be with you here today. We encourage you to make the most of today's experience, interact with the subject matter experts, ask plenty of questions, and attend those sessions that interest you. Also, you may network in person online, and we hope that you learn all that you can today. This event does offer a YouTube live stream, so please feel free to send any of your colleagues that would like to join in that link as well so that they can learn along with us today. Now, this slide provides QR codes for different ways that you can connect with SBIA on social media. We'll also put up this slide during our breaks on screen. You can use your camera on your cell phone to scan the QR codes. And we do encourage you to join us so that you can stay up to date with our upcoming events and our most recent posts. Now, let's turn our attention to some logistics that applies to today's attendees. In the pod, you'll notice that there's going to be a link on the side of your screen to complete the different various session evaluations. These are only available during the individual presentations, so please take a few minutes to complete those as the day goes on. Our overall evaluation and certificate of attendance for today's session will be included in the thank you email following the conclusion of today's event. Those are sent the morning after the event, so be on the lookout for that. And so then, with that being said, please also note that certificate of attendance is only available upon completion there. You have one chance to download it, so make sure that you do so at that time. When that certificate is downloaded there, you can use it in support of RAPS, SOCRA, ACRP, and SQA credits. Make sure there that you do complete it within the next 2 weeks. The deadline for this is June 3rd. Also, as a reminder, these sessions are being recorded, and the YouTube link for that live stream recording will be posted on our event webpage the day after the event concludes as well. Last, if you are an online attendee, you can submit your questions in the Q&A chat pod. We do encourage you to do so that we have a vibrant Q&A session today. And so make sure you check that out during the conclusion of each session. Now, for our on-site attendees today, again, you can note the name of the on-site Wi-Fi. Again, there's no password. You just need to click the acknowledgement. For the Q&A session, you'll notice 2 microphones here in the middle of the room. When the Q&A session starts, please stand up and stand at the microphone. Feel free to go ahead and stand up even if somebody is currently asking their questions so that we have an understanding of how many questions are in the room and we can gauge our time appropriately there. And also, Please note that we are unable to answer any questions that are application specific. We will only be answering questions as they pertain to today's material, and general questions there as the subject matter experts see fit. As a final reminder, also please silence your cell phones and use the internet sparingly, as that can impact our broadcast quality today. A last reminder too for any in-person attendees today, if you haven't done so, make sure you stop by the registration desk and sign in. We do use your in-person sign-in to assess who needs to get that thank you email at the end of the day. So make sure you put your name on the list that you were here today, okay? And then for the next upcoming event, we do note that we have one coming up on June 11th. This is the Advancing Generic Drug Development Bioequivalence Challenges for Patient-Centric Oral Formulations. If you're interested in this, we do encourage you to join us. My co-host for today is Captain Brenda Stodart, who you heard from yesterday. She is the SBIA Program Director. And so if you'll just a quick moment, thank you, Brenda, for that. And now we can proceed with our first session. First, we will hear from Dr. Adam Fisher, Staff Director of the Office of Quality Assurance within the Office of Pharmaceutical Quality here in CDER. Then we will hear from Captain Cyrus Agarabi, who is the Associate Director for Scientific Program Coordination in the immediate office of the Office of Pharmaceutical Quality in CDER. Please join me in welcoming Dr. Adam Fisher.
Thank you, everyone. It's great to be here today. I'm Adam Fisher. Before I dive into my presentation, I do just want to stop and note how this is really a full circle moment for me. So I attended the REdI conference in the early 2010s. I don't remember exactly what year it was, but I attended it, part of the private sector, if you will. And I was part of a new startup company, and I was trying to learn about how the FDA worked. And I got an email one day that said there was this conference that you could go to learn these things. And I thought, well, that's great, but we're scrapping for every dollar we can get. We didn't have a lot of money to spend. And then I saw that it was free. It was a free conference you could go to learn about the FDA. So I thought, hey, this is wonderful. And so I ended up coming to this very conference and learned so much. And there was two things I really took away from that event. The first was how passionate and knowledgeable each speaker was about their area of the regulatory world. And I think in the moment, I didn't realize how important those areas of the regulatory world were because I was relatively new to it. So I thought they were niche areas. I now understand they were, in hindsight, very important areas of the regulatory world, but they were very passionate and very knowledgeable. And then the other thing I remember is that there were some really bad sports coats there. You know, people in the audience and on stage. And I think we've come a long way in the last decade or so, fellas. So congratulations to us. But let me kind of dive in here and talk more about advanced manufacturing. What I want you to take away from my talk today is an understanding of the full menu of advanced manufacturing options that we have in CDER, either to support you or to support someone you know, and some examples of what we've done in the recent past in these areas. So let me just start by saying, as was shown in my intro, I'm in the Office of Pharmaceutical Quality. And quality is an easy word to say, but maybe a little bit harder to define. And so what we believe is that everyone deserves confidence in their next dose of medicine. And it's pharmaceutical quality that actually gives you that confidence. It assures the availability, the safety, and the efficacy of every single dose. And that's why I'm talking here today. It's not because I love advanced manufacturing or, you know, we're tech nerds and we can talk about cool stuff. It's because we want to make a difference to patient lives, and we want to use that. We want to do that by using advanced technologies. And we do think they can make a difference in patients' lives. They can create better quality medicine. So for many years, we've pushed for a drive towards Six Sigma manufacturing quality in pharmaceuticals. In other industries, this is well established. In pharma, in many cases, folks are still driving to get there. Of very much importance right now, it can be used to reshore manufacturing technologies, can help domestic facilities compete in a global market. And I think traditionally we've seen new manufacturing technologies adopted in the US first before they then eventually become adopted in other countries around the world. You can potentially develop drugs rapidly, so you can speed the development of these therapies. You can prevent drug shortages. Another big reason that we support advanced manufacturing, we've seen that more than 2/3 of drug shortages are caused by quality issues that impact the supply of the product to patients. And by addressing those factors that cause these quality issues, you can potentially avoid more drug shortages. And then, of course, emergency preparedness. We've been pushing for this even prior to the COVID public health emergency, but certainly during COVID, it became very clear that advanced technologies would play a big role in emergency preparedness in the future. So let me talk about how we coordinate our advanced manufacturing programs in CDER, and you're in the CDER track here, so this is the Center for Drugs. Before I dive in too much, I do want to emphasize that other centers have similar programs to support advanced manufacturing, in particular CBER, the Center for Biologics, and we do work closely with them in a number of the programs that I'm going to describe to you today. But let's start with the basics here. What is advanced manufacturing? And you would be surprised how long it took to put together just these 3 bullet points here. But it could be integrating novel technological approaches or using established technologies in a newer, innovative way. And I think this is important because there are some technologies that have been adopted in other industries, manufacturing industries, that may not be considered advanced in those industries, but would still be considered advanced in pharma. And so this using these established techniques in a newer, innovative way, or even perhaps used in pharma in a manner that is different from how they might be proposed to be used in the future. And then applying production methods in a new domain where there are no defined best practices or experience. It's really things that would challenge you and would challenge us. And so this is the full menu of programs that we have in CDER to support advanced manufacturing. And they're all under the central coordination of an Advanced Manufacturing Council. So if we break it out into the different regulatory functions, you have assessment and inspection. And in these functions, we have 2 programs that can support developers. The first is the Emerging Technology Program, or ETP. And then the second is the Advanced Manufacturing Technologies Designation Program, or AMTDP. We also have a robust science and research program, which includes an advanced manufacturing research facility that is new, state-of-the-art. I'll describe it in much more detail later. And then we have a policy function, and this is supported by the Framework for Regulatory Advanced Manufacturing Evaluation, or FRAME initiative. And really all of these pieces come together to provide the legs of the table that advanced manufacturing can sit upon. And the one thing I do really want to emphasize before I start is the importance of the science piece, right? Because when we do science, it isn't just for fun. It isn't just because we want to say something provocative and get into a high impact journal. We don't want to have a big press release. We don't want to win an award, but we do the science because we want to use the science to make regulatory decisions better and to make better regulatory policies. In many ways, it's the foundation of all of it. And so as you can imagine, there are some coordination needs across all of these programs, and this is what we do with the council. We coordinate information and data sharing, our strategies to communicate. Part of that strategy is me being here today to talk to all of you in the room and also online. We have strategic alignment, so we have priorities that apply across all of the different functions and certainly needs and expectations for the different functions. And then there is resource, right? We need to address the resource issue. So scanning the horizon for new technologies that are being developed, and then understanding how we can use the resources available to us to the most value to the patient across all of the programs. And so this is what we try to do at a central level. Now let me dive in and talk about each of these programs in a bit more detail. And I'll start with the Emerging Technology Program, which is our oldest and most popular of the programs, which I may also call ETP. So the Emerging Technology Program, it is a collaborative program that was established in 2015 where developers of new technology can come and meet with the FDA to discuss technical and regulatory issues, and this is the important part, prior to filing a submission. Because if we wait to address these issues until you filed a submission, in many cases, it's already too late. We need to meet and we need to talk ahead of time. And this could be related to a product or manufacturing design and development. And specifically, the bread and butter of the program is focused on submission content related to an emerging technology. So that is in a regulatory submission, an NDA, ANDA, BLA, or application-associated DMF, or IND. And the key feature here is that there's coordination between the emerging technology team and the FDA quality assessment team that will eventually handle the regulatory submission. And I can explain a little bit more about how that works, because this is the secret sauce that provides the value in this program. And so in the ideal state, the same emerging technology team representatives that meet with you prior to submitting your application to work through these issues will be the same composition in the review team that will handle the submission and the whole process of the submission once it's received. And some people think, well, a submission's just, hey, you get some paper and you look at it and you check the boxes and you say yes or no, but it's much more than that, right? So it spans the early engagements that we do in this program and maybe beyond this program through controlled correspondence or other pre-submission meetings. It can include an emerging technology site visit where we take FDA staff to your site to learn about your advanced technology before you provide a regulatory submission. Then there is the integrated quality assessment, which is when we put together a team of assessment staff to assess the application and drive toward a decision. And related to that, there's also the pre-approval inspection. And pre-approval inspections aren't indicated for every application, but for many applications, and especially many applications with emerging technologies, they are indicated. And part of the pre-approval inspection will also be connecting with the emerging technology team that is informed about the technology and aware of the discussions that have gone on to that point. So you put all of these things together, and it's a very powerful force for new technologies. And as I like to say, a technology can't remain emerging forever. At some point, it has emerged, and we have a program where we develop sufficient experience with the technology such that we can graduate it from the emerging technology program. And what graduation means is that we are comfortable enough and familiar enough with the technology in order to trust our standard quality assessment staff to handle the submission. And by the way, part of that requires training in order to ensure that that can be done, but this is what it means to graduate from the emerging technology program. And it really is a life cycle approach where we see a new technology, we become comfortable with it, and eventually we train people to handle it. That is what we're supposed to do long term. And in several cases, we've been successful. So we have graduated technologies, including continuous direct compression. This is for solid oral drug products. Newer ones include perfusion with multi-column capture, and then also the implementation of simple Raman models in manufacturing. So all of these are graduated technologies that we have trained staff on to handle now. So a big question we usually get, well, what does this emerging technology space look like? What are the technologies that you see? And so this shows through the end of last year, the technologies that we've accepted into the emerging technology program. I think everyone will probably have the same initial observation. There's one bar much bigger than the others, and that's continuous manufacturing. In many cases, that was the first technology that came to the program, even going back to prior to 2015, 2014, and so on. People were developing continuous manufacturing. But I hope you also see that there are many technologies on there, right? Analytics, aseptic technologies, container closures, distributed manufacturing, which continues to grow in popularity, modeling, simulation, machine learning, novel dosage forms. That's another thing I want to emphasize. Emerging technologies don't have to be only manufacturing technologies. They can also be novel dosage forms and then novel unit operations as well. And so all of these things really make up the emerging technology ecosystem that we've seen since the start of this program. And so then the next question that I get is, well, how many things have you accepted, right? And so this is accepted, meaning the regulatory submission was approved, right? So this is the number of approved applications that have been associated with emerging technology program activities. And for the most part, you see the same initial observation of the prior slide. There's been a lot of continuous manufacturing. There have been some new analytics, container closures, ML, a unit operation, and still growing in some of the other areas. And I want to emphasize, it wasn't just accidental that these technology acceptances led to these technology approvals. A lot of work went into that. And so it took almost 200 ETT meetings in order to get these approvals and 26 site visits in order to get these approvals. So 25 total approvals. So, I'm sure everyone can do the math, but it means about 1 in every 8 ETT meetings is turning into an approval, which I think is actually favorable if you look at the rate of conversion from a new product IND to an eventual approval. So we're potentially above the rate of converting a new product into an approved drug on the technology side. And I have to say, one of my favorite parts of the program, and I hope if you're not familiar with it, one of your favorite parts of the program will be that it's very easy to apply, especially from a regulatory standpoint. So you can start early in development. You don't even necessarily need to have a drug candidate identified yet. You can approach us early. There's a guidance out there for everyone to follow that describes what you need to do in order to request participation in the program. And then my favorite part, and I hope your favorite part, is that it should be less than 5 pages. We're not looking for an encyclopedia here. We're just looking for the most important information that we need to determine whether we're going to accept the technology or not. And basic questions in there. Describe your technology, explain why it's novel, describe how it improves products, summarize your development plan. And this is an important one, implementation roadblocks, and then describe your submission timeline. And I really want to emphasize the implementation roadblocks piece because I do think that's an issue that people sometimes overlook when they turn in their proposal, because the ETP exists to help you work through these roadblocks. And sometimes the application just says, hey, we've got this great technology, you could do these cool things. What do you think? Will you accept us into the program? But if you haven't identified, these are the issues that we need help with in order to be accepted, then it's difficult to justify accepting. And sometimes we have to do a second round. So anyway, just keep that in mind. And then the decision, the goal is always to make the acceptance decision within 60 days. So relatively quickly, and it doesn't always take 60 days, of course, but relatively quickly, we will take a look at it and decide whether we accept into the program or not. Okay, so let me talk about another similar but different program. And this is called the Advanced Manufacturing Technologies Designation Program, again, AMTDP. And this came directly from legislation. And I think perhaps that's one reason that the name is so long and not easily rolling off the tongue. But it came through the FDORA Act of 2022. And so you can see there are a number of sections, and one was the creation of this program in particular. And this is a program that I will emphasize is not specific to CDER. CBER is also involved, but CDER certainly is. And there are some benefits of getting the Advanced Manufacturing Technology designation. So you get timely advice and engagement. You get a lead to facilitate the quality assessment. There's coordination with the assessment team, and then we can prioritize interactions on the advanced manufacturing technology. And this also has its own guidance out there that you're welcome to read. And criteria for eligibility here. This is one I really want to emphasize. So there's 3 main pieces. There's novelty, there's manufacturing process improvement, and then there's drug quality. So for the novelty requirement, you need to show that it is a novel technology or an established technique used in a novel way. For example, something for which FDA has limited assessment or inspection experience. Then the other one is a manufacturing process improvement. So you need to show that the technology will result in a decreased time to market or a supply increase or maintenance for shortage or critical drugs, or there's an opportunity to show that there's another substantial process improvement. And then third, and I'd say this is the one that we have seen the most problems with, is equivalent or superior drug quality. So comparative data for existing drugs that shows equivalent or superior drug quality. So data, right? Not a paragraph that says, oh yeah, we'll have equivalent drug quality, don't worry about it. But data that shows that for existing drugs, or if it's for a new drug that we haven't seen before, sufficient evidence that we'll have equivalent or superior drug quality to another manufacturing technology. So keep these in mind. Again, process is pretty straightforward. You can submit requests to this mailbox. Make sure you include all that data that we talked about for eligibility. The process for acceptance here is a bit longer than for the emerging technology program, so 180 calendar days from receipt. And then, of course, I hope it goes without saying that submission of a request does not guarantee that you're going to get the designation. And I think this is one reason that in CDER, at least, we've seen most manufacturers lean more towards the emerging technology program than the advanced manufacturing designation program. Because in the designation program, the bar's a little higher, the acceptance decision takes a little longer, and so most folks have relied on the emerging technology program. But a question that I often get is, well, what's the difference? You know, how do I know which one to pick? And based on what I just said, I think for many folks, the emerging technology program may be the best solution. But in theory, the AMTDP is for more mature technologies where you have this drug-specific data where you can show equivalent or superior quality. On the ETP side, we can begin our engagements much earlier, as I mentioned, even before you identify a drug product. For an AMT holder, they may be less likely to be the applicant than an emerging technology program participant. For example, if you have a manufacturing technology that could be used across many different types of processes or for many different applications. ETP, as I mentioned, covers not only manufacturing technologies, but also new dosage forms. And then the AMTD, so the designation itself, is an official statutory designation. And we've seen some folks on the CBER side get the designation and sort of immediately throw it up on their website and say, hey, look, we've got the designation. And then one thing I do want to emphasize is that you shouldn't apply to both programs, right? It should either be an ETP path or an AMTD path. We shouldn't apply to both. But a tech ineligible for the emerging technology program may qualify for the AMTDP and vice versa. And just one example on here is if you have an emerging technology that maybe doesn't have the data yet to qualify for AMTD, that can still be fine through the emerging technology program pathway. So lots of options on the table here. Now, let me talk about another one that's very important to me. This is the framework for regulatory advanced manufacturing evaluation. This is something very near and dear to my heart. I started this program several years ago now, and I've since become the chair of the emerging technology program, but I'm still very active in FRAME. So this focuses on the policy framework that supports the implementation of advanced manufacturing. And really, the story of how this began was you go back to the early days of the emerging technology program, and I believe everyone envisioned that we would be having these technical conversations and answering technical questions from people who are developing new technologies to prepare us, again, to receive the submissions. And I think what we saw over the years was that was maybe about half of the questions that we got. The other half of the questions were about policies. And how can I do this within this existing policy? Or how can this policy change? What is this guidance telling me to do? And so because of that, we realized, well, hey, we may need to create an initiative specifically to focus on the policy piece. And then somewhere in the middle, we also had funded this workshop, the series of workshops, actually, at the national academies that produced this report that explained the manufacturing innovations that were on the horizon. And a lot of these new technologies brought policy issues, potential policy issues that needed to be addressed. So 2020, we created this FRAME initiative to really support these new technologies and policy space where needed. And if you really break it down, there's 4 things that we do. We seek and analyze input. So we have public workshops, we have discussion papers, we address risks. So we try to understand exactly what the risks are of a new technology and maybe how you can address risks in different ways from people using traditional technologies. We seek to clarify expectations. This is really the traditional regulatory policy work where we create rules and we create guidance and so on for people to follow in order to implement new technologies. And then very importantly, and I'll talk more about this in a bit, we harmonize it internationally. And you bring this all together and we hope to create a cohesive regulatory framework for drugs. We can't focus on every single possible technology. It's a recipe for failure. So we have prioritized certain technologies for policy focus. One is distributed manufacturing, which I mentioned earlier. This is a movement away from large fixed brick and mortar manufacturing locations and to more geographically dispersed and centrally controlled manufacturing operations. The second is end-to-end continuous manufacturing. Continuous manufacturing is where you have a process that has a flow of material from one unit operation to the next. And we have seen continuous manufacturing of drug substances, and we have seen continuous manufacturing of drug products, but we have seen relatively little in the end-to-end continuous manufacturing from start to finish the whole way through, raw materials to drug products. So that's what end-to-end continuous manufacturing is. As I describe it sometimes, continuous manufacturing on steroids. And then also, I hope this goes without saying, this needs to be a policy focus here, AI. And in fact, I will say that the explosion of AI a few years ago caused us to change our strategy significantly and divert some of our resources that had been focused on certain distributed manufacturing developments and had to focus them on AI. And there's been some good examples of why that was successful. So I just want to run through some of the things we put out. We had discussion papers and public workshop on distributed manufacturing. That turned into our action plan and a stakeholder feedback paper that we put out about a year later. Then, as I mentioned, we diverted a bunch of resources to artificial intelligence in drug manufacturing, had a discussion paper and a workshop. Somewhere in between there was also internationally harmonized guidance released on continuous manufacturing that did include information about end-to-end continuous manufacturing as well. And then the last few years have been particularly productive. So last year, we had 2 draft guidance come out. One was on considerations for complying with 21 CFR 211-110, which I realize is like a foreign language to most people, but a large part of it is about using process models as part of a manufacturing control strategy and how you might do that using advanced technology described in this draft guidance. And then the other is AI to support regulatory decision-making. And this was a guidance that was much bigger than just manufacturing, but we certainly contributed a good bit of manufacturing content to that guidance. We provided a summary of the feedback that we got on AI and drug manufacturing in the workshops that had happened in the past. And then a recent development, very proud of, is that we published, ICH published a reflection paper that we initiated on advanced pharmaceutical manufacturing and how you might work together as international regulators in order to enable this. And I just want to pause for a second on that and just emphasize that international harmonization is essential to domestic manufacturing. It's not something different, right? And I think when people hear that, sometimes it's confusing, but the reason that it's important is because US manufacturers that want to use advanced technologies want to sell their products all around the world, typically. And so if the US is the only regulator on board with the technology, the value proposition on their side is relatively limited. So they like confidence that many international regulators, at least in the major markets, will be okay with the technologies before they implement them. That is why international harmonization is so important for domestic manufacturing. And on that note, we do a lot of work in collaboration with our colleagues in the European Union. We share our knowledge. We sometimes give parallel scientific advice to developers. We share our guidance and policy with one another. And then in some cases, we even have joint publications. We were lucky enough to visit the EMA a few years ago. I'm somewhere, I think they stuck me way in the back, so you probably can't pick me out of the picture there, but I am somewhere in that picture. And it really was a wonderful trip where we learned so much from each other and then made plans to continue to work together in order to make things even better. And one example of that is we came together and we wrote a joint technical publication on process models and model risk frameworks for pharmaceutical manufacturing. And I want to emphasize one thing here too. When we talk about AI, I think sometimes, in many cases, we forget that the foundation of AI is modeling, right? So we need to address modeling in addition to just AI, right? And so this is really looking at the whole concept of using modeling. And so we had authors from CDER, we had authors from CBER, and then we also had authors from EMA who came together to put this publication out in 2024 and describe our joint understanding of the state of using this type of technology. And I will say, I think this was a very rewarding thing to work on because it is now leading directly to the policy conversations that we're having about how do you handle AI models? What is an appropriate level of risk? What is an appropriate level of documentation and so on? And also, I would say an indicator that, at least for me, I've always committed to communicate more than just through guidance. So one of the feedback notes that I got early on was that people felt that FDA only communicated through silence or guidance, and that was it. And so I made a commitment that we're going to do our best to communicate technically, not create policy in journals and stuff like that, right? But to communicate technically outside of just guidance when it comes to new technologies. So this is an example of that I'm very proud of. And then this all turned into this reflection paper that just came out this year that really, at the heart of it, describes a strategy where global regulators can come together and follow a stepwise path in order to create harmonized policy related to the most important advanced manufacturing technologies. It's out there on the web. Everyone is free to look at it. And I'm hoping we can put it in play very soon. Paper just came out. It came out like a month ago. And then it's no secret, there are policies that are publicly out there on the CDER agenda and elsewhere. So one is an establishment registration requirement rule for distributed manufacturing. There's also a draft guidance on CDER's agenda on meeting CGMP requirements for distributed manufacturing, and then a related guidance on application content related to distributed manufacturing. So again, if you remember our prioritized technologies, one of the first technologies was distributed manufacturing. You're starting to see some of the fruits of that labor. Okay. So I'll go very quickly through advanced manufacturing science and research. As I mentioned, this is the foundation of all of it. We have an intramural research program and an extramural research program where we provide grants and contracts for experts to do research in the space. Internally, we have 38 active projects. This spans drug substances, complex products, 3D printing, biomanufacturing, AI and modeling, a bunch of publications, a bunch of reports, a bunch of presentations. Similar extramural research, fewer projects, of course. Significant amount of funding has gone into this since 2019. A lot of it related to continuous manufacturing, digital twins, knowledge management, a bunch of publications, some reports, and 120 presentations. But the thing that I really wanted to emphasize to you all, as I mentioned earlier, was this advanced manufacturing research facility that we recently got online. So this is the first facility related to drug advanced manufacturing research. It's 8,000 square feet in Ammondale, Maryland. It's a reconfigurable research space, so sort of a mini ballroom facility, if you will, where you can roll unit operations in and out. It supports and bridges research policy and assessment and provides a dedicated training space for FDA staff. So this is where folks can come in and learn about these new technologies so that they can handle them in regulatory submissions. And we're looking forward to using this to inform policies, guidance, standard development, and so on, and train even more staff on these new technologies. Some of the research programs that we're targeting there include continuous manufacturing of drug substances, and then also solid oral drug products, but then also biological products. So it can handle chemistry and it can also handle biotech in the same facility. And complex drug product manufacturing, just as one example, we have lipid nanoparticle manufacturing that you can do within the facility that is super cool and a lot of potential there as well. All right, so I talked a little earlier and I just sort of breezed through, there's a bunch of presentations and there's a bunch of papers and stuff like that. But I think where the rubber meets the road is the impact of this research. So the first impact of the research is that we have the scientific experts contribute to the recommendations that we provide through the Emerging Technology Program. So they're scientifically informed recommendations. Beyond that, then there are regulatory actions that we actually take. And so the research has directly informed 43 different regulatory actions. So that has provided the scientific basis of those regulatory actions. It informed policy, so ICHQ 13, which I mentioned earlier, also ICHQ 5A. And even resulted in an award for some of our staff through the American Institute of Chemical Engineering for their work in developing models for quality risk management in continuous manufacturing. And it's a wonderful paper. It lays out how you can use these models effectively in the manufacturing space. So I just want to close here with one case study, because I described a number of different programs, and I want to illustrate how it's supposed to look when it's done successfully. And for that, let's start with continuous manufacturing, because that is where it all began back in 2015. So continuous manufacturing in general is great for processes with high repeatability requirements, which I'm sure we'll all agree describes pharmaceuticals. It may not be fit for every drug manufacturing process. I want to emphasize that. It may not be fit for everything, but for some things it is. And in those cases, it can provide better efficiency. It can reduce the costs, reduce your footprint, and also improve process control. And you can see here, so this was in our paper, just between 2016 and 2020, continuous manufacturing of drugs went from almost nothing to several $1 billion worth of sales through the implementation of products from different companies. And what this looks like is a conversation that happened between the FDA and the experts who are developing these technologies. So we were told that manufacturers were hesitant to adopt continuous manufacturing without additional engagement from FDA. So we created the Emerging Technology Program in 2014, 2015. And then manufacturers said that they weren't comfortable until FDA could show that they approved a product manufactured with continuous manufacturing, which we did in 2015, and since then, again, 17 more. Then manufacturers were hesitant to switch. A lot of batch manufacturers out there, they're hesitant to switch without FDA showing that they could approve a switch from a batch to a continuous process. We did that in 2016. Then we heard it would be hard to adopt CM without guidance from FDA. So we put out the FDA guidance in 2019, quality considerations for continuous manufacturing, and then also related this draft revision to viral safety evaluation of biotechnology products, also had some elements of continuous manufacturing. Then we heard they're hesitant to adopt CM because it might take longer for FDA to approve their products. And in 2022, we did a self-audit and we published a paper that showed that continuous manufacturing applications were actually approved faster than similar applications that use batch manufacturing. And then finally, they needed internationally harmonized guidance, and that came out in 2023, which I already mentioned, ICHQ 13. So this shows, again, how it's supposed to work. We have a conversation, we address pieces one step at a time. And I just want to focus on that self-audit for a bit, because continuous manufacturing applicants had shorter times to approval and marketing compared to batch applicants. So 3 months faster to approval when compared to similar applications, and 4 months faster to marketing when compared to similar applications. We also looked at some other potential regulatory barriers. So the difficulty of making manufacturing process changes or difficulties in pre-approval inspections. And we also didn't identify any substantial barriers there either. So I think it's a good story, one that still has more development. We have 17 approvals of continuous manufacturing. I'm sure we'd agree there's probably room for more, but I think what's there is there. So in closing, just want to say, let's talk. You're the expert on your technology. We want to hear from you. And we don't know, or we may not know unless you tell us about it. There's some resources for anyone who gets these slides later, and thank you so much for your time.
All right, thank you for that, Dr. Fisher. So at this time, we will open up for questions. Again, if you are online, we encourage you to put that in the chat pod. In room, you can go ahead and line up at the microphones. We do ask that just for the sake of time with these Q&A sessions, you do consolidate your question to a single direct question for our subject matter experts. We also request that you refrain from any personal commentary or answers, just so we can focus on getting that technical guidance from our subject matter experts today. So with that, it looks like we do have a question in the room. Go ahead.
Thank you for the presentation. I just wanted to ask a question on that 17 approvals for continuous manufacturing. Is that across different companies, or is it one company is doing the heavy lifting?
It's across different companies. Now, some companies do have multiple products approved with continuous manufacturing. Some of that's in our paper that came out in 2022. There's a graph, you can see some of that. But it has been distributed among different companies.
Thank you for that. If at this time, if there's anybody else in the room, please stand up and come to the microphone. I'll keep prompting as well. Anybody? Okay, if not, are there any questions online for us today? Oh, looks like we may have a question for in person. If you would like to go ahead and come to the microphone, please.
It was a nice discussion. I was wondering if there is any case study of where it failed actually,
Where
these discussions failed to, and you thought that this is not the process should be allowed to.
where discussions will fail. Okay, yeah, fair enough. I think that an important thing to clarify here is that the Emerging Technology Program exists to address the questions that folks have about developing new technologies. It doesn't mean that they always get the answers that they want, of course, but it's to provide clarity, developmental clarity for how they can be developed. I'm sure there's some cases over the years where it's maybe not been as successful as participants hoped. But I will say at one point we did an exit survey of participants in the program, and they had rated their satisfaction rating an 8.9 out of 10. Now, we're a government agency, we're a regulatory agency. We don't get rated 8.9 out of 10 on anything, okay, right? So I think clearly we're making some folks happy out there, but not always. And I'll say another thing that has surprised me as I've become the new chair of the program is seeing how many companies we accepted into the program and never heard back from. Surprising, because people seem interested, they seem gung-ho, we accept them, and then we never hear back. It's actually a surprising number of companies that fit that mold as well. So yeah, there are reasons that it may not be successful, but I think by and large, it has been a successful program.
Thank you for that question. At this time, if you'd like to line up, we'll go ahead and take some questions from online to balance here, and then we'll come back to questions in the room.
So one question we have, since 2015, I'm so used to speaking loudly, now you can hear me properly. Since 2015, how many new emerging technology applications did the FDA receive, and how many technologies got the ETP designation till date?
Okay, so the technologies that had the ETP acceptance, that was on my slide. Off the top of my head, I don't know what those numbers add up to, but I'm sure someone could pull them up and add them up. I want to say it's between 100 and 200. And then the first question was the number of submissions that we'd received.
And yeah, how many did we receive, and how many actually got the ETP designation till today's date?
Okay, so how many people requested to be in the Emerging Technology Program versus how many were in? Yeah. Off the top of my head, I don't know the exact number. I will say that the majority of people that request have gotten in. I think it is relatively rare, but I think I mentioned some of the reasons that we may turn it down, either because the technology has not been shown to be novel, or maybe more often that they've not identified potential technical or regulatory hurdles that they need to work through with us. So there certainly are some, but I think the majority we have accepted. I mean, the program exists to engage with people and to help them. We legitimately want people to be successful. Of course, we also want patients to be safe, more importantly. And so we try to work with people when we can.
Thank you very much.
Thank you for that response. We'll go ahead and take our next in-person question.
Yeah, unfortunately, my question is along that line. And as a matter of fact, I mean, what I have seen that is ETP accepted, for example, aseptic technology is probably 24, but then ATP approved is 0. So I really like to know that is what actually went wrong, and there's what we need to really develop in that front so that, I mean, it can be approved.
Sure, and I think the one bit of data that's not evident in my slide is when we received the submission, right, or the request, actually. So, for example, if we had a request for an aseptic technology that we received in November, there's not yet enough time to be in an approved submission, right? And then sometimes the development timelines are just longer. As I mentioned, you can come to us before you've even identified a drug product, maybe before you've even identified a manufacturer that wants to use your technology, to have an early conversation. And so sometimes it takes a while to develop the business, beyond just the technical aspects of it, but to develop the business. And I do want to emphasize too that we will have repeat engagements. It's not just a one and done deal. So sometimes people will get feedback from the program. They'll go back, do further development, submit another meeting request a year and a half down the road, and we'll have another engagement, and it will eventually lead to something bigger. But I do also want to emphasize that sometimes the ultimate goal is not necessarily evident by an approved application. An example might be the use of an AI system, for example, that you might use at a manufacturing facility that might not be in a regulatory submission per se, but could influence regulatory submission, certainly an inspection of that facility. So that's an example of a technology. We may have multiple engagements on the technology. It might not ever end up in a submission. It doesn't mean it wasn't successful. It just means that wasn't the end goal. So there's more to it than just the numbers show. Maybe I'll just add my commentary that I do think aseptic technologies are very important and a key area for future development. So I would encourage everyone to consider the program if you have something that might fit that area.
Thank you for that response. I'll go ahead and again encourage anybody in the room to line up at the microphones if you have questions today. If not, we will go ahead and take another question from online.
Okay. Thank you very much for all the great information and putting together this slide deck. Those are the person's comments, not mine.
Well, I'll thank Ruth August on my staff for putting together the slide deck. She did a wonderful job.
As the head of the ETP, a place where people can submit AI applications for review, is there a technical definition, for example, of what CDER considers to be an unacceptable AI system, preferably in the generative AI space?
Well, this one is a little tricky. The guidance for ETP is about a submission, right? And so I do think there are some AI technologies that could be promising, that could be advanced, but might be so far removed from an application submission that it might not make it into the program. An example, just off the top of my head, if you had, let's say, a technology for supply chain tracking, that could be very powerful, but it probably wouldn't get in the ETT because there wouldn't be a direct connection between issues that we need to address in order to have a regulatory submission. So there could be some examples where it wouldn't fit. Though I do want to be clear, we are open for meeting with people who are developing AI technologies in manufacturing, and we are doing so right now.
Excellent. Thank you for that. With no other questions, we can go to another online question.
Sure. Here's a pretty simple one, I think. May a company have more than one submission to the ETP?
Yes, many do. Yeah, and there's many companies that we've engaged with over several years and then have submitted another technology that we have picked up and engaged with them through a separate ETP pathway. And yeah, it's actually fairly common. And I think once you're successful once, and especially once you understand the value of the program, you're more likely to come back and do it again.
Thank you.
Of course, and final call for in-person. If not, I think we have time for one more question.
One more question. Would an ETP company be advised to move to AMT by ETP when applicant would be a good candidate?
That's a good question. It's admittedly not something that we've run into before. I think on the CDER side, the benefits are relatively similar. We would engage with you, we would have meetings, we would answer your questions, we would try to make a connection to the assessment team for your submission. So if pressed, I would probably say if you're already in ETP, it would be easier just to stay there and maybe not as beneficial to transition to getting the designation, although that may be a business decision as well that may be outside of FDA's purview.
Excellent. Well, thank you so much for your responses today, Dr. Fisher. If everyone could give him a quick round of applause.
Thank you.
Excellent information. Now at this time, we will go ahead and welcome Captain Agarabi up to the podium for our next presentation today. We'll be presenting FDA PreCheck Pilot Program, a CDER Perspective. Please join me in welcoming Captain Agarabi.
Thank you. Thanks, Corey and Brenda, for putting this together and inviting me to speak on this topic. In case folks join late, I'm Captain Cyrus Agarabi. I work in the Office of Pharmaceutical Quality in the immediate office. And today I'm going to be talking about the FDA PreCheck Pilot Program from a CDER perspective. I really hope that through my talk, I can really introduce this new pilot to folks both in the room and online so that they get an understanding of how we develop the ideas and where we're trying to go with this new program. By the end of this talk, I'm confident that folks will at least understand the basics. All right, so today I'm going to give you some background about what the driving force to initiate this pilot program and the environment that really had a call to action to start looking more seriously at domestic manufacturing and onshoring. I intend to give you an overview of the PreCheck pilot program and how we envision it to operate in the future and the various different aspects of the program, as well as updates on the actual implementation and roadmap of the program. And finally, looking ahead where we see the program launching and going successfully into the future. So some learning objectives as people are getting continuing education. By the end of this talk, we hope that you'll understand the policy foundation and manufacturing challenges driving the PreCheck program. Learn about the two phases, what we're calling the facility readiness is phase 1, and application submission is phase 2. Reviewing the eligibility and selection criteria for the pilot program participation and track and implement our implementation milestones through the pilot launch. And finally, we plan on discussing current progress and stakeholder engagement like we are today, engaging with our stakeholders. So just to take a step back and look at some data from 2025, this is CDER specific for products that we regulate. And the numbers paint a very particular picture, right? So as of 2025, approximately 53% of branded drug products are manufactured outside of the US. About 69% of generic products are manufactured outside of the US. And only about 11% of active pharmaceutical ingredients, or API, manufacturers are in the US, compared to 22% in China and 44% in India. And Adam actually talked about some of the impacts of supply chain challenges and the need for domestic manufacturing onshoring from a perspective of advanced manufacturing, but it's also critical for all kinds of manufacturing, whether it be advanced or traditional. So in response to these, the situation, there was an executive order, which I'll refer to as EO 14293, that was signed about a year ago. And it was aimed at regulatory relief to promote domestic production of critical medicines. The takeaway was, how do we maximize the timeliness and predictability of agency review while streamlining and accelerating the development of domestic pharmaceutical manufacturing? So in response to that, we came up with the PreCheck program. And the goals here are exactly that. How do we build regulatory predictability to support US pharmaceutical manufacturing capacity, right? So what are our strategic initiatives and priorities? Capacity and resilience. Through this program, our goals at a high level are to increase domestic capacity. And why do we need more capacity? Well, less drug shortage. Patients need drugs. Drug shortages cause problems. Global supply chains can have risks, right? So we're trying to mitigate those risks. How do we do that? Well, decreasing time to operational readiness for new facilities is our strategy. In order to get more new facilities online, the PreCheck program aims to move us to a different paradigm for this pilot program. It also supports adopting innovation. So it's timely that this presentation follows Adam's presentation on advanced manufacturing, right? There are technologies out there that are going to help new facilities come online faster. We have innovative strategies to make smart facility decisions and really leverage that information. So that's definitely a piece of the scalable solution. So we're really looking at how do we get the biggest impact for our efforts here. So before we even started the program, I think it's also really helpful to understand, see, the impetus was we're going to start this program, but we really wanted feedback. So we put out a Federal Register notice last August. And in the Federal Register notice, we invited industry and stakeholders to come here September 30th for an in-person and hybrid meeting. We had over 1,800 people online and about 45 people in the room. It was really important for us to get industry feedback on our approach and see what they thought was going to be valuable and see what they thought that wasn't. And I'm going to talk a little bit more about the public meeting and in subsequent slides. We then had a docket review that was also attached to the Federal Register notice, where we also were able to look at more feedback and really distill the key pieces that were going to be valuable for folks in this program that we can do. We launched a website on February 1st. This created an application portal, questionnaires and eligibility criteria were provided, and folks had in this program. Internally, we've been working very hard to develop. We're able to invite the folks who had previously expressed their interest to provide detailed information and more substance to their proposals for evaluation. Right. So I'll talk a little bit more about timelines, but this just gives you an overview that before we're starting to launch this program, we really are trying to engage in stakeholder outreach, get feedback, and see what's going to be useful both for the agency and for the stakeholders involved. So again, that September 30th meeting was really, really pivotal for us. We presented our framework, we gathered the stakeholder input on the tools and the program applications, and we had two tracks. The morning tracks was focused on finished dosage forms, and the afternoon discussed active pharmaceutical ingredient manufacturing. This way, relevant stakeholders could be participants in the areas that make sense, and we could be more focused in those discussions. We had representation from CDER, CBER, and OII from the FDA side, and we had a cross-section of manufacturers, including new drugs, generics, biologics, and CDMOs, with over 45 in-person attendees and over 1,850 online. It was a moderated discussion on PreCheck implementation strategies. It was a very robust conversation. I think we built in time to have sort of engagement, and it wasn't just they ask a question and we respond. In a lot of ways, it was meant to have multiple sort of back and forth to get down to the core, the core pieces here. And I think it was a really good conversation that we were able to have. Piggybacking that, the docket, which is the official record for comment on a new program. If you're really interested, it's public. You can go to see FDA 2025 N-2489. We got the feedback and went through every single comment. And in general, the feedback was positive. And it was really about clarifying. And I think part of what we're trying to do here today is continue to clarify, continue to set some level of expectations. But really, how are we going to prioritize participation? What are the implementation timelines? So this is, last fall. What are the clear expectations of industry? If we're going to participate, what do you want from the industry side? How much do we need to be involved in, and how much resources is this going to take from us? Also requests for scope expansions and considerations. So we definitely considered the feedback, and we ended up moving forward in a two-phase approach. So shifting gears to give you just a high level based on that feedback. The first phase of the program for the pilot is envisioned to be a facility readiness piece. And the key here is very similar to the concepts Adam was bringing up about early engagement, right? So if we can talk about things earlier, focused on facility manufacturing information, so not product dependent, but around the facility, we can have these conversations before operations start. So the earlier we are, the better chance we have to have an engagement and make meaningful feedback. And we can do this through what we're referring to as pre-operational reviews, which I'll discuss in much more detail, and the use of facility-specific drug master files. When we get all this information, we have this engagement, we're going to move to a second phase. And the second phase is really about tying it to a submission, right? So at the end of the day, products get approved through this pathway, right? We don't just approve a facility to make whatever, it's tied to a submission. So engaging in pre-submission meetings to resolve issues and expedite assessment of the facility-related manufacturing information, whether it be an NDA, a BLA, an ANDA, or a supplement, and to support the future inspections involved. So we think key program benefits are about FDA collaboration and engagement. Doing this during the initial facility development phases, not tied to a product submission, is very, very unique, right? Many of the other programs we offer here are very much tied to specific product. An enhanced communication throughout the facility evaluation process, including the pre-licensure inspection and pre-approval inspections, and earlier feedback that ensures continuity with later facility evaluations, including the facility inspection, right? So we want to have early engagement that carries forward. And I'm going to talk in more depth about that. So I'm going to take a sip of water because this is a complicated slide. My goal here is not for us to go through each box. It's to give you a sense that the first phase is not linear. It is sort of synergistic. So we talk about, we initiate, people get into the program, and we've got these two pieces of the puzzle. We have a pre-operational review, which I'll get into the specifics in subsequent slides. But for this slide, these are going to work together, right? So we're going to be able to have engagements, right? So wherever you are in your facility design and build, we're going to be able to have engagements. We're going to talk about things like your construction, your equipment, your IQ, your OQ, your PQ, your validation. We can talk about all these things, whether that's in a POR, whether that's through a meeting-driven approach, whether that's through a POV, which is in the later phases of element 4, where we come and do a site visit. We're going to have a lot of different tools to get this information. And putting it all together is a type 5 DMF. Because what it's going to allow us to do is create a central repository for this information. And it's a living document. So as the facility goes, you can see the arrows move. I have a pointer. As the facility gets further along, right? So we start here where let's say we're just starting out. As we're going along, we're building this knowledge base. We're having these interactions. We're giving feedback. And then it can live in a DMF so that we make sure that when we move forward, we have this information in a central repository. This is meant to be synergistic, not duplicative, right? So being able to use these two pieces together in tandem is going to get us, we hope, to here. And the goal at the end is facility readiness. Can we say this facility is ready to move to the next step? We have looked at their systems. We have done a site visit. We have looked at the DMF, and we feel, we would recommend. Now, if there are issues, there are going to be feedback, and there are going to be reports that say, we would encourage you to address these gaps before you move forward. Because what we don't want to do is have someone move too fast and then get themselves in a situation where they're not quite ready yet, right? So this early engagement before the submission allows us to have more time to do that. We'll also say this figure and many of the other figures are on our website, so don't worry about pictures. But really, we think the early benefits are going to be, having this engagement before all the capital is deployed, in case you need to pivot, in case there's some issue. It's good to sort of identify things early. Facility readiness summary report for phase 2. We're going to be able to take this information and bring it forward, right? It's not just going to live in one place. It's going to move forward with the program. And finally, a product agnostic facility information is in a centralized DMF. And I'm going to talk a bit more about these specific pieces because not everyone is familiar with PORs and DMFs. So the other piece is understanding that there are expectations for participants with this in mind, right? So we do expect and encourage folks who participate to open and update a Type 5 DMF throughout the facility development. We want them to engage in pre-operational reviews through written communications, meetings, and FDA directed, whether it's on-site or remote visits. We do want them to submit facility documentation, and this can be our design plans or equipment qualification, and respond promptly to FDA inquiries and meeting requests throughout the process. So it's really meant to be a dynamic two-way communication as we move into this pilot program. So what is a POR in this program? We've sort of broken it down into sort of 3 boxes, right? So early on, you can think about we're building the building, right? We need to know what the water systems are. We need to know what the facility design is. We need to understand early engagement so that we can provide feedback on the facility designs, the pharmaceutical quality systems. Building in quality up front is a strategy that's going to help us down the road, right? Having these conversations to identify where there could be gaps early and is going to help us as we move through the process. And this could be, more likely going to be written communications or meetings for check-ins and a meeting package-driven approach, right? Where we're going to focus early, equipment design, PQS, personnel planning, nuts and bolts, for lack of a better term, right? As we move forward, we're going to move into a later element. The facility has been built, equipment has been put in place. How are we going to, how is the facility going to be run and operated, right? So we're going to verify, our goal is to verify the facility is constructed and equipped according to reviewed designs. Did you do what you said you were going to do early on? Is it still the same? Did things change? Things do change sometimes. Not significant deviations would be good. This can look a little different where we can do some on-site visits. Remote tools may be utilized here, and a written summary report after those visits could be really valuable for both sides to understand what's working and what might need some improvement. And really, we're focusing on physical verification, data reviews, procedural and systems audits, and outcome reports for readiness. So the ultimate goal is to have a ready facility, right? So we want to get people onshore, and we want them ready quicker than they normally would be. So the outcome here is a successful completion of the verification provides confidence. It's going to provide confidence for this facility. It's going to provide confidence for the agency in their state of readiness. The final POR outcome report feeds directly into subsequent application pre-submission meetings for specific products to support their quality assessment. Also, a lot of this information, as I said, is going to end up in the DMF. So we do these meeting packages, we do the site visits, we do all these different steps, but there's so much involved when you're standing up a new facility. Wouldn't it be great if it lived in one place that could be referenced and cross-referenced multiple times? So this is a concept called qualify once, reference many. So as we open the DMF in the pilot, it's going to be very lean, for lack of a better term. Early on, if you haven't built much, we're going to find out about what you plan to make. Where is it located? But as we go through the POR, it's going to grow, right? So each of these elements is a kind of a milestone. So we see a pre-construction phase where relevant information. Construction, the thing is built. What do we need for that phase in the DMF? So we start to populate and we grow. And by pre-production, we're going to have things, covering everything from the layout design, your qualification, your validation, equipment information. What's your HVAC? What's your water, right? Facility utilities and environmental monitoring, pharmacy quality systems, knowledge management, CAPA, deviation strategies. How are you going to do contamination control? None of this is necessarily product specific. type 5 DMF is not for product specific, but it's facility specific. All the things I just mentioned have an element from the facility, right? We then incorporate that DMF by reference into future applications so that when you do have an application, you do have a product, you can reference it back. And you only, hopefully, will be putting in the relevant pieces that are going to be product specific here. That's going to save us time. It's going to save the participant time, right? And the other thing is, there's going to be additional facility content, and this is a living document. There's going to be a need for amendments. There's going to be a need for maintenance. But once we get this built in this pilot program, it's going to be a very valuable tool moving forward. So we have completed the basic concept of phase 1. All of that is going to move us into phase 2. And really the idea here is we take that knowledge forward. So when we go to a pre-submission meeting, we're going to go and look back at that DMF. We're going to go back and look at that POR site visit and say, hey, these are things that need to be addressed. Were they addressed? Are there new things that we need to be aware of? Doing this before the filing, doing this before the submission gives us time because we're going to know more about that facility, more about when they plan to manufacture in case we need to do an inspection while they're manufacturing. It's going to help us plan. It's going to help the facility plan. So I think there's a lot of benefits here to utilizing this information, leveraging those engagements, and moving through the normal process, right? So after the pre-submission meeting, this is all pretty standard, right? So you're gonna submit something, whether it's an NDA or a BLA or an ANDA or a supplement. The benefit here is if we've gone and looked at a lot of the GMP aspects or more general stuff, we may be able to do a more focused PAI or PLI on some of those extra pieces that weren't covered, or if they need to be revisited. And this is going to help us to make some decisions earlier, potentially, in the cycle. So if we know what we're looking at and we know more about the production schedules, it might help us to plan earlier in the cycles and avoid some of the communication challenges we may face. Our hope is to, whether it's favorable or not favorable, we can't guarantee that. We can't guarantee favorable outcome. We can say that our goal is to have more information and potentially earlier engagements to decrease risk, right? And that's what we're trying to achieve here. So yeah, I think that is the general idea of phase 2. And our expectations for our folks participating in the pilot are to let us, work with you to participate in the facility-focused pre-submission meetings to support accelerated facility evaluation, right? The more we know about your plans when you're ready to, a bit before you submit your application, the better we're going to be able to react to that and to engage with that. And also, keeping your GMF up to date so that you can reference the relevant facility-specific Type 5 DMF in the submitted marketing application. Those are really key expectations, as well as just general engagement strategies throughout the pilot. All right, I got 8 minutes, so I'm going to talk to you. I think we've covered the benefits through engagement and feedback. But I think taking a minute to talk about where we are on the roadmap, right? So this came out in January, and it follows this timeline for launching a pilot. And February 1st, the website became live. Folks could fill out an evaluation form and let us know they had an expression of interest. By March 1st, the window closed, and we reviewed the submissions. By April 1st, those finalists from the first round that were going to be invited to do a second round were notified and were given additional instructions of what they needed to provide. May 1st, there was a deadline for them to submit requests for final application. And we are currently in this phase. And June 30th is coming real fast. So we will be completing our evaluations of the full proposals and with a plan to notify on June 30th for the final pilot program participants. And I'm stuck. Thank you. All right. So Who is eligible? You have to be a new manufacturing facility, a domestic manufacturing facility that is not an existing facility or an extension of an existing facility. You have to be located in the US. The PreCheck pilot program is limited to new manufacturing facilities located in the United States or its territories. Facility size threshold. It needs to be a facility that produces sufficient volume to meet a specified market need. It doesn't mean you have to make X millions of units. It means if you have a small volume, it might be fine as long as it meets the need of the patient population. And that could be the case for a rare disease state with a limited population. Or it needs to significantly contribute to addressing US market supply needs. So I think it's finding the right balance of the type of facilities that can meet these needs. Finally, you have to commit to actually submitting an original NDA, ANDA, or BLA, or application supplement for the new manufacturing facility during the PreCheck pilot. Additionally, a commitment to keep that product in the United States for 3 years. What we did not want was to put a lot of investment in, something to go through and then be outsourced. Would defeat the purpose. All right, priorities, right? The source of the materials is actually really important, right? Because when we're talking about supply chain resiliency and onshoring, it's not just I'm a facility that makes X, right? There's an integrated pieces here. There's KSMs are key starting materials. There's if I'm a finished dosage form manufacturer, where do I get my active pharmaceutical ingredient from? If I'm a API manufacturer, where do I get my key starting materials from? So where they get their US-based materials from or not was important because that ties into the whole strategy. What they're making, and from a CDER perspective, you know, we cover small molecules, we cover certain biologics, peptides and oligos and other therapies. So I do think from a CDER perspective, looking at the range of products and understanding that this pilot we're trying to learn more from and having a good mix is important. Also, if they make a product that is on the shortage list or an essential medicines list, those things would probably be pretty useful. What is the facility development timeline? You know, we want to balance facilities that are in the earlier phases where we can have an impact, but we don't want that timeline to be so long that we sort of miss the window of opportunity here. So timeline was dependent on the development stage and expedience to market and innovation. You know, dovetailing on the ETP type of discussion, if they've got innovative solutions like modular or prefabricated, advanced automation, data and digital integration, these are things that are, you know, good to have. Finally, company qualifications. Can they do it? Do they have the experience, or do they have the partnerships? Do they have the necessary expertise, or can they get it? We really want folks to be successful in this program, and we understand that that takes the ability on both sides. All right, looking ahead. So, as I said, we talked through that. Final selection notifications will be issued June 30th. The pilot is expected to launch shortly after notifications are received. And all industry stakeholders should monitor the PreCheck website regularly for program updates and future cohort announcements, if possible. So what are we going to learn? Getting back to our learning objectives to wrap this up. The pilot program will be used to test, learn, and refine our approach across key success areas. Supply chain resilience. We're hoping new domestic facilities supplying critical medicines and reducing foreign supply-driven shortages. We're looking to gain regulatory efficiency, both through reduced facility development to approval timelines, and early communication can help us reduce downstream risks. The pilot lessons will inform scalable models across product types and company sizes. Can we replicate this? Can other facilities replicate their learnings from this pilot program? And I think I've covered this pretty well. Just a key distinction that PreCheck engagement is proactive. We're hoping issues can be identified and discussed before the submission stage, reducing our downstream delays. And this is a recap and resources. So we do have a great website. I strongly encourage folks to go to the website. It not only gives you background about the Federal Register notice and the program, how it's being stood up, but we also added many of the figures you saw in the presentation today to the website in sort of a program overview to give folks a better understanding of what we're envisioning this pilot to operate in that model. There's also an email box if there are questions related to PreCheck. And with that, I'd like to thank my colleagues across CDER, across CBER, and OII, as well as the Office of the Commissioner. It's been a really good group working together collaboratively to get this pilot moving forward. With that, I can open us up to questions.
All right. Thank you so much for that presentation on the PreCheck program. We will now open up the floor to questions. I'm sure you guys know at this point on day 2, if you're in the room, please line up at the microphones. We will go in the order that we get them. If there's anybody in person, please line up at this time, and feel free to line up also during the entirety of this Q&A portion. We'll go ahead and begin with the first question in person.
Good morning, Captain Cyrus. Thank you very much for the presentation. The first question is regarding the finalists. I think I already attended the last month's REdI conference as well. I know there'll be 9 finalists that'll be selected eventually. Will that be made public?
That's a great question. We are still discussing the ins and outs of the communication strategies. So I'm going to decline to answer right now because, yeah, the communication strategy may change. I think at a minimum, June 30th, the facilities that are accepted are going to get notification that they're in the program. And that is, from sort of the SME technical side, that's where our focus is in our group, because we are different than sort of the external communication folks. So our plan is to make sure that the folks who are selected are notified on June 30th that they're invited and what the next steps are and more information.
Thank you. And one follow-up question to that is, did you guys rule out the packaging facilities completely from this, or is there any consideration in case if it's a high volume product where X company would like to, put a packaging facility in the US, will that also be considered into this program where we can start, engaging with the FDA?
So I can say the current, submission window has sort of closed. I would point you to the website for future engagements. I think these are all ongoing discussions and learning opportunities for the pilot program to see if we expand and where we might expand into as we move forward. So the short answer is it really depends on who's applied. And, we're taking all the expressions of interest and evaluating them based on that eligibility criteria and the prioritization criteria. So, yep.
Thank you for that response. It does look like we have another in-person question at this time.
Yeah, I have potentially 2 questions. So just to follow on from that, is there any consideration for continuing the pilot after this initial set?
Can we start first and then?
Yeah.
I get this question a lot, and there's a lot of enthusiasm. And my short answer is, if we can be successful here and gain support, that will help us as we move forward. It is hard to commit to what happens after pilot launch because pilot launch has not happened yet. And I think there's going to be a lot of learning. And I think it is a wonderful opportunity, we also intend to get a lot of feedback from the participants to see what's working and if things need to be modified or not. The short answer is everything sort of depends on resources and priorities. And we can revisit this once we've sort of launched and got our feet under us and see where the resources and priorities are at that time.
So it may not be a one and done.
I cannot comment on that
Okay.
because.
Second question, this might not actually be your question. I noticed that you had on there innovation and facility design. The previous presenter didn't really have any in the ETP program. So where do you see maybe some of that innovative facility design, that modular design fitting in?
So as a former ETPer, I can say maybe his slide didn't capture that, but I can tell you there's work in this area, and they're known to us through the ETP and other places. They we engage. So I think there's a synergy here. And I'll also comment that if something is so innovative, the PreCheck program isn't going to step on the ETP's toes. Adam and I sit, down the hallway from each other. We're gonna talk about stuff. So I think understanding the synergistic opportunity here is really important, and also being able to coordinate these pieces together is really going to make the program valuable. And if there are folks who are interested in that or have those capabilities, again, part of the priorities, if it's going to help have a high quality facility onshored faster with more predictability and less risk, that sounds like a pretty good strategy for the program we're talking about.
Thank you.
Excellent. Fantastic response. At this time, we'll go ahead and take some online questions as well.
Okay, the first question is, does this program apply to producing vaccines and oncology drugs and also pediatric medications?
So, as the title said, this is from a CDER perspective, but as I have acknowledged my CBER colleagues, I can say at a high level, it's not indication based, it's modality based at this point. So as long as you're in the product jurisdiction of CDER, those would be potential products of interest that could be in the program.
All right. Excellent. Again, if there's any in-house questions, please feel free to line up at the microphone. If there are no in-house questions, we can take another online question at this time.
Certainly. Does a new facility mean a new manufacturing building needs to be built, or can a company buy an existing facility for new use?
Can a company buy? So it's like new to you, like a used car? Sorry, I'm trying to get that one. What do you think, Adam?
I think the question is basically, could a company buy a U.S. facility to start manufacturing there? And by doing so, would they be eligible for PreCheck?
It needs to be, I think this gets to an important point. The goal is to have an engagement while they're developing the facility, whether they're purchasing it outright or they're building it themselves. So, buying something that's operational already would kind of defeat the purpose, as opposed to saying, okay, we're going to buy this land and build this building and so on. So hopefully that answers it. But yeah, really, the earlier on in the construction, the more opportunity for engagement, the higher the value of this program.
Excellent. Thank you for that. It does look like we have an in-person question today.
Thank you, Captain Cyrus. Just a quick question. Is there an anticipated duration for the pilot?
That's also a good question. We're going to have to see. I think, I don't know who we're going to end up with yet. And it's going to depend on timelines. And some folks might be a little further involved and some might be others. And there may be some hiccups, right? And roadblocks when you're building something new. So the short answer is, we're committing to participating and getting through the phases, and that those timelines are going to be more tailored to what the need is for the specific participant.
Thank you.
Thank you for that. Looks like we do have time for one more question today. So if there's no in-person ones, we can take an online question at this time.
During the pre-operational review process, how does FDA distinguish between the early POR elements focused on design and qualification concepts versus the late POR element focused on physical verification?
I think that's a good question, and I want to say that the elements are sort of meant to be guidelines, right? Things can be probably over the guy, depending on how things are being built, and it may not be quite as sequential as this, right? It might be a little more fluid. So I think really when we're talking about the earlier phase items, it's as I said, kind of the nuts and bolts and getting the facility built and getting the underlying frameworks. And as we get further along, the facility is going to start running, even if it's not making approved product. It's going to be doing things, right? And that's where going for a site visit to go and see what is the PQS in practice, what does their environmental monitoring in practice look like, that's where it starts to change into the later phase to really be able to understand the facility readiness. So I think it's a spectrum, but it's something that we're going to be able to, as we engage, differentiate and use these sort of as milestone markers as they progress.
Excellent. Well, I thank you so much for that fantastic information about this program. If everyone will give me a quick another thank you to Dr. Fisher and Captain Agarabi. They will be present on site during the break. We'll go ahead and adjourn now for a 15-minute break. The Sodexo kiosk will be open for any snacks or any coffee that you may need. And also our speakers will be present during this time, not the lunch break, but right now if you do have any follow-up questions or discussion points for them. We will resume at 10:50 for our next presenters, and I'll see you then. All right, welcome back everybody from our break. I hope everybody had a quick little time to reset before our next sessions today. So first, we will be hearing from three people from the Office of Digital Transformation who will be presenting information on FDA electronic submission updates to the eCTD for version 3.2.2 and version 4.0. First will be Heather Crandall, an IT advisor for ODT. Next, we will hear from Jonathan Resnick, and also an IT advisor for OTD, as well as Seyoum, Senay, the Supervisory Operations Research from ODT. And then our next presentation for the morning session will be verifying data reliability, observations on bioavailability and bioequivalence, and good laboratory practice studies supporting the development of new and generic drugs and therapeutic biologics from Sean Kassim, Director of the Office of Study Integrity and Surveillance within the Office of Translational Sciences. Please join me in welcoming Heather Crandall.
Okay, hi everybody. So my name's Heather. I am here from FDA to talk about CDER submissions and updates to the eCTD for both versions 3.2.2 and 4.0. So our FDA disclaimer. So I'm going to get into a little bit about what's new, an update on our current status on the 4.0 implementation, and then a little bit on some of the common errors we've seen across both versions of the eCTD submissions. So this one's a little bit of an older update, but I want to revisit, bring this to everyone's attention in case you guys didn't see this. Back in February of 2025, we had 23 new values for file tags if you're referencing version 3.2.2, or keywords if you're referencing version 4 of the eCTD. We had 23 new values added to that for granularity on study data. So if you want to know more about where and when we recommend these new values be used, there's information in the Study Data Technical Conformance Guide linked here. Also, we had some new file formats added to our specification for studies. So just wanted to raise that, make sure everyone sees that. We also have added some new validation criteria to our specifications for both versions of eCTD. We updated a handful of the 3.2.2 validations, including 2024 and 2025, as well as added a new code 2026 that have the effective date of TBD. 2026 is a medium level error. You can see some of the snippets of that specification here, and we added some equivalent error codes for version 4 with the numbers listed here. Again, effective date TBD. As usual, we will give notice before when these are going to be effective before they are made effective. So everyone will have that time. But, you guys can go in, take a look at the language. Feel free to ask eSub any questions that you might have around the language. But again, we will let you guys know before they are made effective with plenty of time. An update on our implementation of version 4. So last year, we rolled out eCTD version 4 in FDA, started accepting new applications in version 4. And we have gotten some. I'll get into the numbers on that in the common errors that we see. What are we working on right now? We are currently working on forward compatibility and implementing forward compatibility. So we've started a pilot, a small pilot with some PDUFA members to test out software and make sure, see how everything's looking. Again, we will let people know when we are ready to start accepting forward compatibility. So this would allow current applications that are coming in that 3.2.2 version to start using 4.0, being able to lifecycle, reference, reuse, continue their applications using now the 4.0 standard. So we will let everybody know when that is made available. We may not put it on the data standards catalog because we are already accepting 4.0, but we will certainly put it out with lots of notice on the web page. So, but 4.0 is currently listed on the catalog, linked here, along with all of the other, the other version of eCTD and all the other standards and terminologies that FDA currently supports, requires, all of that. So some of the common errors we've seen for eCTD, I tried to focus on what we've seen across versions for this. So for 3.2.2, and I looked at all of 2025. So not for 2026 yet, just the past year. Something to keep in mind when you're looking at these numbers, we had over 300,000 submissions in 3.2.2 format, and the errors account for 1.7% of those. So very small amount in comparison to the total number of submissions we received. Just keep that in the back of your head as we're talking. But we do see things like duplicate sequence number. This is a common one from year to year where either a sequence number is accidentally reused in a next submission, or a sequence is accidentally submitted to us multiple times. Maybe a button is clicked twice. So that's a common error that we see. I have listed our validation numbers from the specification that you can reference. 1306 tends to be high on the list every year. That has to do with well-formatted leaf references in your 3.2.2 backbone. So something to keep in mind as you're looking through this. For 4.0 submissions, we actually did receive 39 4.0 submissions last year, and 74% of those submissions had errors. Now, it's a small number to begin with. This is a new version, so there are going to be some errors as people are learning this, trying this for the first time. If you have any questions about an error you might receive while submitting in 4.0, please reach out to eSub Reject, all of this. We are happy to help answer any questions that you have about any errors that you're receiving, any rejections for 4.0. Also keep in mind that we have sample submission. You can do sample submissions to us before you start submitting formally in version 4 if you want to work through some of your questions that way too. But again, we do see duplicate sequence numbers, missing or incorrect forms. These are all common errors that we see across both versions of eCTD. So breaking down the duplicate sequence numbers a little bit more, since this is very common. Remember, the sequence number must be unique, and there is that difference in how the sequence numbers are formatted between 3.2.2 and 4.0. So 3.2.2, it must be 4 digits. It has those padded zeros at the beginning, whereas 4.0 does not have that requirement. So instead of submitting sequence 0001, you'll just submit sequence 1. And with forward compatibility coming out, this will also hold there. So you might be at sequence 0003 in your 322, and then you would submit just sequence 4, no padded zeros, to continue that application in version 4. So that unique sequence number would still carry across through the forward compatibility. And then, just keep in mind the fillable form, since that was another error across both versions. So we're seeing either a fillable form is not included, maybe an incorrect, the wrong kind of fillable form, or maybe the form was no longer fillable by the time we received it. It was flattened, so we can't read the data out of it. Just something to keep in mind as you're putting your applications together. These required forms that must be fillable. So I know I went through that rather quickly, but if you have any questions, you can reach out to eSub. And there are other links to other information that you can reference. And I will now turn this over to Jonathan. Thank you, everybody.
Great, thank you, Heather. All right, so I'm here today to talk to you about one of FDA's demonstration projects that we've done as part of our PDUFA 7 commitment. One area we wanted to look at was leveraging cloud to see what we can do about creating a more efficient, standardized process around how these information requests are created, sent from the FDA to industry, how industry can track these, and then send them back in. So that's what we're going to talk about here. So what is an IR? Just in case anyone needs a quick little refresher there. FDA issues an IR when reviewers need additional information or clarification on that application. And the review team can issue an IR at any point during that review cycle. And there can be multiple IRs going on under a particular application at one time, which makes it all the more important to be able to have centralized tracking of what's going on with these. So the overall big picture with our IR modernization effort is we want to establish a consistent, standardized, and secure collaborative framework that's going to improve that communication between FDA and industry. So we set out to do this by working collaboratively with industry. And what we did is we worked through our PDUFA 7 quarterly industry meetings that we have with pharma and bio. And we work with them to find a list of companies that we could work with so that FDA is not working in a silo, all on our own. We wanted to actually hand in hand talk about what are the challenges that industry is having, and what are some things that we could work on in a short period of time in this demonstration project. So we split it out into initially 3 sessions back in 2025, where we had a kickoff meeting. We talked about the issues that FDA has on its side and the issues we think industry is having on their side based on a questionnaire that we put out ahead of time. We came back session 2. We prioritized the top challenges. What could we move out on in the next few months to actually create something that everyone can look at and react to? And then we follow that up with session 3 back on May 6th in 2025, where our team was able to actually demonstrate something back to our industry partners. And we did get a favorable reaction to it. And there were some things that were unfavorable, and we worked together to iron those things out. Then a year later, February 10th, 2026, just a small typo on the year of that last column, 2026, we actually came back and built something in our pre-production environment where our industry partners could actually log into the system and get their hands-on experience. Actually, they sent in a test submission. We sent back a test IR. They were able to use this this platform that we had to try it out and give us their feedback. So that's kind of to level set everybody and what we were doing. So there are three main areas that we set out to improve. One is having a secure portal delivery interface. So we wanted to provide that single secure portal where all the IRs can be accessed, reducing the risks of miscommunications. Why is that important? Because what's happening today is if you're working with CDER, you might get an IR via email, you might get it via fax, you might get it in the postal mail. If you're working on an IR with OGD, you're probably going to get it as an email, and it's an attachment to the email. And you're getting multiple IRs on a given application number. And so it becomes very difficult to track all of these. And many times when you're responding to an IR, you might not respond to everything in that IR letter, in that amendment you're sending. You might spread it out over a course of several amendments. This all just stacks on top of itself and makes it difficult to track. So this secure portal delivery interface and what we were using is the CDER NextGen portal, a portal that my colleague Seyoum Senay is going to talk about in just a minute that is already up and running in production that many in industry are already familiar with. And then we get to the next column there, sponsor reviewing and responding. We set up an IR tracking dashboard and an interface. So as a sponsor, if you own 4 different application numbers, when you log into that CDER NextGen portal, you are going to see all 4 application numbers, and you're going to see whether or not an IR has been issued under each one, and you can click on it. Say you click on application number 55555, and there are 3 different IRs. Well, you're going to see them all in one spot. You'll even see the status of it. And then finally, in that third column, FDA receiving and reviewing these IRs, we wanted to improve the process in how FDA can automatically map these responses to the IRs against the originating questions that came out to be able to automatically match that up. Because today, the review offices are spending a lot of administrative time just trying to match up the response to the questions that went out. So this is a high-level view of just the overall flow. We tried to kind of not get too complicated in this so that we don't lose anybody. So blue is FDA side, green is industry side. So the idea here is the FDA review office within whatever CDER app they're working on. And today we're working on CDER One Nexus is what we're working on. So within that tool, they would be able to, as they're reviewing the application, log in their particular, questions or additional piece of information they need. And then the project manager of the file, right from that same tool, can automatically load those into that formal IR letter. When they send it, you go to step 2, which is green, where the IR actually goes to the CDER NextGen portal, and the sponsor gets an email notification that lets them know, hey, CDER has issued an IR against this particular application number. Click this link to go into the CDER NextGen portal where you'll be able to view that IR and interact with it. And then once industry is done interacting with the IR and doing what they need to do, they then submit their response as they normally do. So I have a couple of screenshots of what we did here. So we have our secured portal delivery interface. That's kind of what it looks like. So you come in from an industry standpoint and you can see your different application numbers. You can click on any tile and it'll give you all the IRs underneath it. Your IR tracking dashboard interface. So for every single IR that's issued to you, you can track which items have you responded to. If FDA on its side has changed the status on any of those items, it would flow through to you so you could see it, which helps with the transparency aspect. And then also within that CDER NextGen portal, as a sponsor, you're able to create what's called an IR response file. It's a structured data file that contains key pieces of metadata, like the IR letter unique identifier, the unique identifier for each question within that IR letter, and that file you would then include in your eCTD submission that you send back through ESG. And that's what this slide depicts here. And that brings to the summary of all of this. And so what did we demonstrate in this proof of concept? We showed that we can use structured machine-readable IR exchanges. It's perfectly feasible. We did it. We built it out in our pre-production environment, and we were able to do this within our existing eCTD ESG regulatory framework, so we can move quickly on it. We demonstrate how we can move to a near real-time FDA industry communication hub, automating those linkages of the IRs and responses, and then eventually move to a point where we have all FDA industry communications being exchanged in that structured way. So I am going to hand this off to Seyoum. He's going to talk more about the CDER NextGen portal, and then we'll have some time for Q&A afterwards. Thank you.
Good morning, everyone. My name is Seyoum Senay. I am a senior program manager here at FDA. Today, I would like to share 4 short stories, and we're trying to focus more of the process and impact. Some of you have been a part of NextGen portal for a while, so I really appreciate Brenda inviting us to keep coming here and present. So we will be delighted to share what we have been doing the past few months. So with that, so the stories will show CDER NextGen portal is transforming the way the FDA provide digital services to patient, industry, and the public through smarter, faster, and more connected digital platform. So as you see, if we have to put FDA process for illustration in one page, this is the page. Submission comes from sponsors, and we call it a data intake system, as you see NextGen portal right there. And then some way, somehow, it's connected to our internal data store, workflow, as well as publishing capability. So the last year or so, we were standing key platforms for the agency as a shared services, and NextGen portal is being one of them. So with that, at the high level, what's NextGen portal? It's a one-stop shop for purpose of non-eCTD submissions, collaboration, and reporting. And the portal continue to reduce burden to sponsors, academia, research institutes, and small business. So part of it, that's why we are here. And I will share a few stories how we put small business as part of our journey in the NextGen portal. So, as I said, I'll share 4 stories. Story number 1, from paper chaos to digital order. So before NextGen portal, on the left-hand side, FDA received industry submission on paper through email. Staff manually sorted them, identified the right office, rekeyed data, a slow error-prone process that delayed reviews and frustrated everyone who was involved. Today, with NextGen portal, sponsors submit through a single digital portal. Data is validated at the entry, clean and complete. Workflow route automatically through the right team. The APIs, application program interface, handle what used to take days or manual work. So it's a complete integration end-to-end, back and forth again, the two-way communication. So what's the result? With that, we have faster reviews and more time to FDA staff to focus on what matters most, evaluating science and review applications. So with that, I will jump into story 2. The story two will be, our products. We have about 26 products in CDER NextGen portal. We categorize it into 5 category. With that regulatory submission, for example, if you take a research IND, any academia or individual researcher can submit and then treat it the same as, any other bigger industry submission. So the power of that is you can build that research IND in NextGen portal. You can take your time, back and forth, and completely digitally transformed. So the next story is protecting the drug supply chain. The Drug Supply Chain Security Act requires electronic training of prescription drugs across the US. The challenge was getting the FDA and thousands of trading partners to coordinate quickly when suspect product appeared. Here's how it works. A trading partner spots a suspect product and submit a form 3911 digitally through the portal. FDA review and, if needed, initiate an investigation, sending a digital workflow back to the partner requesting transaction records and documentation. All electronic, all traceable, no paper. The impact, faster detection of counterfeit drugs. So with that, a simple 3-step: submit, review, investigate. So this process is happening, digitally. Used to take, months, years at a time, but now it's pretty much near real time. The next story I would like to share with you, the catching drug shortage notification before they start. Drug shortages are patient safety issue. The CARE Act required manufacturers to notify FDA early when shortage was anticipated. Since we stand up this portal for drug shortage since 2021, we have processed over 26,000 industry submission, which is structured, validated data. FDA can act immediately. We are no longer waiting for the shortage to make the news. We also opened a public-facing drug shortage portal where patients, healthcare providers, and organizations can report new shortages as they see them. That is a nationwide early warning network, and it lets FDA act before the shortage become a crisis. So with that, 26,000 plus number is a powerful notification. And our analytics capability, since it's end-to-end, it addresses those challenges immediately. So the last story I have is summarizing this. Today, the NextGen portal has 22,000 plus active users spanning from sponsors, academia, research institution, and small business with 26 product lines. A small biotech submitting their first research IND application gets the same streamlined experience as a large pharma. A hospital reporting drug shortage gets a direct line to FDA. Regulatory burden reduced, collaboration improved. In closing, the digital transformation is not a finish line. It's a commitment to continue improvement in service of public health. The NextGen portal has already eliminated paper chaos, secured the drug supply chain, caught shortage notification early, and built a community of 22,000 plus. And then we are not done yet. If you're not yet using portal, NextGen portal, visit our NextGen portal help center, and my team is here to help. And then with that, thank you for your participation.
Excellent. Thank you for that. We will now invite the three speakers up to the podium to begin our Q&A session for today. And with that, if there are any in-person questions, please go ahead. As always, line up at the microphone. Because we have three presenters doing the Q&A, we do request that you direct your questions to the individual presenter before asking. And at that time, we'll go ahead and start. If there's any questions in the room. All right, first call, any questions? If not, go ahead and line up. Don't be shy. We'll go ahead and switch to an online question at this time. Oh, it looks like we may have an in-person question.
Hi, thank you all very much. I guess for Heather, I feel like we didn't react appropriately when you said we don't need the zeros anymore in sequence numbers. Like I wanted to stand up and cheer, so thank you. That's cool. And for Jonathan, I was just wondering because we might change the FDA point of contact and the login to the portal is currently by user. If there's a way that like a sponsor could a user sponsor is able to sponsor users able to access questions for their products across users, or if it will remain specific to a single point of contact. Or
right, yeah. So if you were using the portal and you had a question about, was it access to, you're coming back up, let's see.
Yeah, sorry, just to clarify. So if I'm the point of contact for an IND or any application and I receive questions about that application, but then someone else takes over that program from my side.
Right, so if you have an organizational change over on your side, and someone else is taking over. Right, absolutely. So what you would want to do is you'd want to notify the help desk for that portal. So if you're using ESG NextGen portal, you want to notify the help desk there to let them know that a different person is going to be taking over, and then they can guide you through what that process is. It's the same thing if you're using the CDER-NextGen portal. So step one, I would say, is just contact the help desk, let them know that you're leaving your organization. And if you do know who the new name is, that's even more helpful because then they can help get that whole transition started for you. And of course, that's different than if, a regulatory contact for an application is being switched over. Then there are some different forms that you actually fill out, and you'd find that out from contacting your regulatory project manager on it. But yeah, great question. Thank you.
Thank you both. At this time, looks like we have another question in person. If you'd like to step up to the microphone, please.
Thank you, everyone, for the great presentations. Quick question on the API DMFs portion. I know it's not part of the ANDA as such from an application standpoint, but it's part of it. So would this program be extended into the API DMFs eventually, or what's your take on that?
So are you talking about the IR process that I was?
Yes.
Okay, great. Yep. So if there's an information request related to the API, absolutely. Yeah. Yeah, because what we set out to do is we wanted to make sure we had a consistent IR process that's used whether you're doing an ANDA, an NDA, a BLA, it doesn't matter. That way, from a industry point of view, you're getting a consistent way in terms of how communications are coming to you and how you're communicating back. And so a question on an API is just really, right, it's a question about something in Module 3 of the eCTD application. And so if the FDA, over an OPQ, Office of Pharmaceutical Quality, if they have a question about the API information in there, and they typically ask that question through an IR, yes, the idea is that they would then use this new model that we've been testing out. Now, keep in mind, this is a proof of concept that we've tested out with industry. It's not in production. I don't have a couldn't tell you a timeline for when that will happen. But needless to say, the feedback that we receive from this is quite positive, and it is all being fed into FDA's overall IT modernization efforts. So this information from the proof of concept is going to inform FDA's roadmap moving forward and implementing these new types of things in the future.
So just more clarity to that follow-up is, I'm talking specifically on the DMFs portion as well. Will this be applied to the, I know NDA, ANDAs and everything comes into picture, but DMFs also are part of it.
That is certainly the goal. Yeah, that's the goal of it. Yeah, we want consistency, and DMFs really become part of the application. We know how they're used, but in general, a DMF does become part of that overall application because of its referencing that critical content.
Thank you.
Excellent. Thank you for that. At this time, we'll take an online question.
Okay, I have to say we have quite a few questions online, but I know we're short of time, so I'm going to try to do one question for each person if there's no other questions in the room. So Jonathan, you're there at the mic, so let's start with you. And I hope this question hasn't already been asked. Does the IR response still get submitted in eCTD via ESG?
Right. So does the IR response still get submitted in eCTD through the electronic submission gateway? Yes, it does. What happens is you use the CDER NextGen portal as the place where you receive the IR, where you can view it. If you need to ask some sort of clarifying questions around it, it's also where you can actually indicate which questions underneath that IR letter your eCTD submission is going to address. The portal then generates this structured IR response file, which you then download and include in your eCTD submission that you send through the ESG. Thanks.
All right, thank you for that. Any other in-person questions before we go on with the online questions? Looks like we do have an in-person question.
This question is for Jonathan. So is this going to be like a long-term repository of where the IRs will be stored? Or, because you just mentioned that you still have to go through and submit it another way. So is it just like a temporary hanging zone while for like, a certain amount of time and then it gets, removed? Like what happens?
Right. So think about the CDER NextGen portal in this proof of concept as being used as a communication tool, right? And ESG just being like a post office, right? ESG is just how you transmit your submission into the agency. But CDER NextGen portal is where you have this two-way communication going on, where you can actually do some actions there on that IR. So in terms of long-term repository, really can't get into that at this point. Personally, I see a lot of possibilities going forward. I think we've all acknowledged we've now have moved to this cloud environment. We're in this environment where we're looking at how can industry and regulator work together on an application or exchange questions and answers and information. How can different regulators who are receiving the same product for review collaborate with one another? And so as more and more of these pieces become connected in the future, I think we will move away from that the communication interface is happening in the CDER NextGen portal, but you're actually sending in your submission through the electronic submission gateway. Instead, it'll all be in one place. That's the vision. That's where we're going. And in the meantime, what we're doing is we're testing out some concepts now with what we've got. But thank you. That was a very insightful question.
Of course, and we are at time right now, but they will be present during the speaker one-on-one there, and we will give some information for the in-person attendees to be able to follow up with any questions that are remaining. It's excellent to see great interest in these question sessions. But thank you so much to our presenters for this first panel. And at this time, we'll go ahead and welcome up Dr. Kassim for his next presentation today.
Good morning, everybody. Thank you for having me. Thanks to the SBIA for hosting the REdI conference, which is a fantastic time to get at all the good information. My name is Sean Kassim. I'm here as the Director for the Office of Study Integrity and Surveillance. And I'd like to take a few minutes to walk you through the work that my office and explain how we verify data reliability of applications submitted to the agency. Now, this presentation draws on real-world inspection findings, so I really hope it'll be both informative and practical for those of you working in or alongside regulated research settings. So today we're going to cover three core questions. Why? Why are we here? Well, because good decisions require good data. If we're going to make high-quality approval decisions, we need to be confident that the data supporting those decisions is accurate, complete, and reliable. And then who are we talking about? Well, we're talking about the FDA's Bioresearch Monitoring Program, also known as BIMO. And specifically, one of the aspects of that is a role in my office, OSIS, within CDER. And then what? Specifically, we're going to walk through what OSIS actually does, our BABE and Good Laboratory Practice inspection programs, how we define data integrity and quality, and what we've been seeing in the field through some concrete case examples, and then closing with a call to action. So why does this matter? Let me start with one of our two major programs my office oversees, the Good Laboratory Practice Program. It's abbreviated GLP, but we had that years before the GLP receptor agonists. Good laboratory practice, it's described in a set of regulations codified in 21 CFR Part 58, and they were established back in the 70s in direct response, the 1970s, in the direct response to the documented cases of scientific misconduct in non-clinical research. And the regulations really exist to ensure that the non-clinical safety studies submitted to the agency are conducted under rigorous and well-documented scientific conditions. They define strict controls to ensure that the data from animal and in vitro studies, are scientifically sound and can support critical regulatory decisions. And they typically occur before any human exposure occurs. And the second compliance program that we associate with the Good Laboratory Practice is the Animal Rule. Now, this program, it governs the evaluation of medical countermeasures where human efficacy trials are ethically or impractical. For example, in case of biological, chemical, radiological, or nuclear threats. In those cases, animal studies must carry the evidentiary weight that human trials would otherwise provide, which makes, of course, the quality and integrity of those studies all the more critical. The other major program we oversee in OSIS involves bioavailability and bioequivalence studies, or BABE. Now, these are pharmacokinetic studies that measure how much of a drug's active ingredient reaches the site of action and how quickly. Of course, this information is fundamental because it characterizes how a drug behaves in the body, it informs dosing decisions, and critically, is what's used to establish whether a generic drug is substitutable for its branded reference product. Now, for these determinations to be meaningful, of course, the studies have to be validated and well conducted. The data must be trustworthy. When the studies are conducted appropriately, we can have confidence that patients receiving a generic drug are getting the same therapeutic benefit as those receiving the brand name drug. But when these studies are not reliable, the confidence breaks down, and that has real consequences for patients. Over the past 10 to 15 years, we've observed a troubling pattern. A subset of sites submitting BABE data to the agency has had significant problems with data integrity, including in some cases, outright falsification or fabrication of study results. And these are not isolated incidents. They represent a systemic challenge that has required sustained attention. The downstream consequences are, of course, very serious. When data supporting a bioequivalence determination is flawed or fraudulent, any approval based on that data is built on a false foundation. And given that 90% of all prescriptions filled in the United States are for generic drugs, the public health stakes could not be higher. So let me turn to who's responsible for addressing these issues, verifying these data that are so critical for decisions. Well, at the broadest level, the FDA's Bioresearch Monitoring Program, or BIMO, operates across all the FDA product centers and serves three essential functions: to protect the rights, safety, and welfare of the subjects or participants involved in FDA-regulated clinical trials. That's, of course, of the utmost importance. And then, as I've been talking about, the verification of the accuracy, reliability, and integrity of the clinical and non-clinical data submitted to FDA. And then also to assess compliance with FDA's regulations. All of the rules specifically around consent and ethical review as well. And importantly, compliance is not just BIMO is not just a compliance function. It's a scientific integrity function. Its work is what gives the agency confidence that the data underlying its decisions reflects reality. And as I mentioned, BIMO is not limited to any single center. Every FDA product center and program, drugs, biologics, devices, veterinary medicine, tobacco, and foods, has a BIMO component. And all of these work in coordination with our Office of Inspections and Investigations, or OII, and specifically within the Office of Bioresearch Monitoring Inspectorate, or OBMI, to conduct inspections that carry out these compliance programs. Now, I filled in the names of all the acronyms here on this slide to make sure everyone has that context. Centers are CBER, CVM, CTP, Human Foods, CDRH, and of course CDER, where my office sits. And all these, again, connect to OII, OBMI to execute their BIMO programs. And the specialization of the OBMI investigators is critical to make sure that what we need in the BIMO world gets done during inspections. Now, within CDER, we have two offices that share responsibility for our BIMO mission, and they work closely together. In the Office of Translational Sciences, my office, OSIS, we lead scientific evaluation for good laboratory practice, animal rule, and BABE BIMO inspections. In the Office of Compliance, the Office of Scientific Investigations, or OSI, handles the enforcement and adjudication for OSIS's programs, as well as the clinical investigator, IRB, complaints, and post-marketing reporting inspection programs. My colleague, Emily Gebbia, is going to be giving a presentation this afternoon that I hope you get a chance to listen to. Together, OSI and OSIS ensure that CDER's conclusions from BIMO inspections are thoroughly evaluated, appropriately documented, and effectively communicated back to the review offices that depend on this information for their review decisions. OSIS operates according to a life cycle model that we think of as a continuous circle of 4 phases: select, evaluate, review, and support. We select sites by analyzing incoming applications and study submissions, applying a risk-based model to identify which sites warrant inspection priority. We have a limited resource. We can only inspect a limited number of the sites that need inspection. Now, once we've chosen that, we evaluate the sites through on-site inspections or remote regulatory assessments, RRAs, and that allows us to examine how the data was generated and how studies were conducted. The team of amazing reviewers in OSIS actually could go out to sites and figure out where things have been done, particularly for our analytical portions of the BABE program. We also then review all the evidence gathered. We piece together any of the findings to determine, are there integrity or quality concerns that call about the usability of the data? And finally, we support the agency's decision-making by providing data reliability recommendations to the relevant review offices and coordinating with OSI to ensure that any compliance follow-up is appropriately pursued. Now, this slide provides a little bit more detail in how that workflow operates in practice. Applications of all types, investigational new drug applications, biologics, and abbreviated new drugs or generic drug applications flow continuously into CDER, as we saw from our ODT colleagues. Lots of submissions coming in for CDER. And alongside that, we have compliance information coming in the form of complaints or referrals from other groups. And so these come in through our review offices, the Office of Clinical Pharmacology, that's also in OTS, the Office of Generic Drugs, the Office of New Drugs, and the Office of Pharmaceutical Quality. All of these generate the ANDA, BLA, and ANDA workload that drive OSIS's inspection programs. Now, this information feeds into OSIS, where we apply our risk-based site selection model, determine if a site needs to be inspected. Our staff in OSIS go out or direct OII, OBMI folks to go out, evaluate the sites, collect any inspection findings. Those findings come back to OSIS, which we then review, assess if any compliance actions are warranted. Those go to OSI, and then provide reliability recommendations back to the review divisions to make sure that they can support their approval decisions. And now we get to the what. So what exactly is OSIS doing when we conduct these evaluations? Well, at its core, we're confirming that the data supported to support these critical applications is actually usable. And we do this through three interconnected concepts. Data integrity is focused on, are the data complete, consistent, accurate? Importantly, integrity failures are not limited to intentional misconduct. That's really, really bad, but poor data management can often undermine integrity just as surely as deliberate falsification. Data quality asks whether the data are fit for their intended purpose and sufficient to support the analyses and interpretations being requested. This means assessing whether the data was collected in accordance with applicable protocols, SOPs, and research standards. And then finally, data reliability is the overarching conclusion. Does a site's overall handling of data and study conduct provide adequate assurance of both integrity and quality? When the answer is yes, we can recommend the data for use in regulatory decision-makings. When it's not, we cannot. And over the past decade, FDA has issued a number of public communications, including letters to specific research organizations indicating data integrity failures that rendered their BAB work unacceptable for regulatory purposes. And these communications are publicly available on FDA's website and serve as an important signal to industry. They're not hypothetical concerns. They reflect real findings from real inspections, and they've had real consequences, including rejection of applications, and in some cases, the need to repeat entire studies. The link here should go to that page and allow you to look further into that. Now let's take a look at some of the examples that we've identified. So our first example comes from the good laboratory practice space and involves misrepresentation of roles. Under 21 CFR Part 58, good laboratory practice regulations establish a detailed accountability structure and for non-clinical safety studies. Every party involved in a study who collected the data, who analyzed the samples, who performed which procedures must be accurately documented and disclosed to the sponsor and then ultimately to the agency. And in this example that we've encountered, a site was listing its own name on studies that were actually performed by outside contractors. So by doing this, the site obscured the true identity of who conducted the work. The sponsor had no way of knowing which entity was actually responsible, making meaningful oversight impossible. And this goes to the heart of what good laboratory practice is designed to prevent, a submission that does not accurately reflect the chain of scientific responsibility. And this isn't just a paperwork issue, it's a scientific accountability issue. If we can't determine who did what, we can't evaluate whether the study was done correctly. Our second example involves informed consent in clinical BAB trials. So informed consent, of course, is the ethical foundation of all clinical research. Before anyone can participate in a study, before they can be exposed to an unapproved drug, give blood samples, or contribute their time, they must be fully and clearly informed about what the study involves, what risks they may face, and how their participation contributes to the broader scientific goal. Then it's not optional. This is a legal and ethical requirement. And in the case we're highlighting, a site included ambiguous, contradictory, and downright false information in its informed consent form, or the ICF. And it fundamentally undermines the consent process. A participant can't give meaningful informed consent based on a form that is misleading or internally inconsistent. Clinical investigators must treat ICF development with the same rigor they apply to study protocols. The form must be clear, accurate, and complete, and failure to do so doesn't just create a regulatory deficiency, it represents a fundamental breach of the trust that study participants place in the research enterprise. This example involves a pattern that we've identified in bioanalytical data that raises serious data integrity concerns. On the left side of the figure, you'll see individual test to reference, or T to R, ratios for Cmax, the peak drug concentration, across multiple study subjects. In a valid bioequivalence study comparing generic to reference product, we'd expect that these ratios to cluster around 1, with the 90% confidence interval falling between 0.0 or 0.8 and 1.25 as required by FDA's bioequivalence standards. On the surface, when you look at the cumulative average across all the subjects, the data appear to meet that standard. But when you examine the data more closely, breaking those subjects into 2 sequential cohorts, a very different picture emerges. So the first cohort of subjects shows T to R ratios that fall consistently below the 0.8 lower bound, while the second cohort falls consistently above that 1.25 upper bound. So the 2 cohorts are pulling in opposite directions, and their averages happen to cancel out to get you the answer of 1. But this is not normal distribution of biological variation. This pattern suggests something fundamentally went wrong, whether it's manipulation of samples, improper analytical handling, or other misconduct, such that the combined result is not a valid measure of bioequivalence. Now, what makes this pattern particularly concerning is that it would not be visible without close, granular examination of the data. That's precisely why we do the work we do. Our fourth example addresses what you'd call overly flexible processes, a pattern where insufficient documentation and inconsistent adherence to SOPs create significant data integrity and quality concerns. When a site repeatedly rejects and reruns analytical samples without adequate justification, or when a rate of repeated runs is unusually high, this raises an important question. Is the site testing into compliance, running samples over and over again until it gets the result it needs, rather than accurately measuring it, what is actually there? Similarly, when SOPs are routinely disregarded or when exceptions are made without proper documentation and rationale, this signals a breakdown in quality oversight. SOPs exist precisely to ensure consistency and to create a verifiable record of what went on and why. A site that doesn't follow them, or that follows them only when convenient, cannot assure the agency that its data is reliable. SOPs are not just bureaucratic checkboxes. They're a documented scientific logic of the laboratory, and when they're bypassed, the scientific logic breaks down. Example 5 focuses on data security and access control failures, what we have characterized as uncontrolled systems. The specific issues we've encountered: free access to rooms designated as controlled, meaning there's no meaningful restriction on who can enter spaces where samples or instruments are stored. Open access to study samples, with limited or no traceability regarding who handled them and when. Uniform access privileges across different levels of staff, making it impossible to reconstruct who was responsible when anomalies occur. Audit trails showing simultaneous data entry by the same individual on different machines, a clear indicator that credentials are being shared improperly used. And then source data being overwritten during backups. This destroys the historical record, makes it impossible to reconstruct the original data. When access is uncontrolled and records are incomplete or overwritten, the agency loses the ability to independently verify what happened during a study. That renders the study and the data unreliable by definition. Data security is not just an IT concern. It's a scientific integrity concern. The chain of custody of data must be carefully maintained as the chain of custody for physical samples. They're important on both sides. Now, our final example is involving self-reported site problems. Situations where a site become aware of an issue and choose to report it to the FDA. Now, FDA genuinely encourages early reporting. When problems are identified, there's an opportunity for dialogue and for finding solutions before they become unresolvable. And we really want that conversation to happen. But for early reporting to be effective, it must be complete. The site must clearly identify the relevance to FDA-regulated work so that the agency can appropriately scope its response. And all relevant details must be disclosed upfront. When critical information is withheld, and we don't get the whole picture, and as we've seen in some cases, the agency cannot conduct a full and fair evaluation. The result is wasted time, wasted resources, and a much more prolonged and burdensome process that would otherwise have not been necessary. It's important to remember, partial transparency is not transparency. If you're going to come forward with a problem, come forward with the whole problem. That's the only path to a real resolution. So all these examples lead naturally to sort of a set of questions that we find useful and revealing when evaluating a site's quality systems. And I'd encourage everyone in the room, whether you work at a site, sponsor a study, or oversee a CRO, consider how confidently you could answer these. Can you fully reconstruct the study as it was performed? Who did what, when, and why? Are roles, access privileges, and change controls clearly defined and actually enforced in your systems? Are audit trails complete, secure, and routinely reviewed for anomalies? Is your staff properly trained to follow the record-keeping procedures, identify problems, and escalate them, importantly, without a fear of reprisal? And is there meaningful oversight that can catch problems, drive improvement, and verify that corrective actions actually work? If any of these questions give you pause, that's exactly where you want to focus your quality system investments. So in letting me distill what we've seen across these examples into some concrete best practices. For the analytical side of things, stability is so important. So we like to start with that. Stability data has to cover all sample storage conditions and handling steps throughout the study. It's one of these main pieces. But in general, the next one, documentation. It has to be sufficient to allow reconstruction of the study, including decisions and deviations. Process and procedures also across all of our programs, your SOPs, and your protocols, they must be clear, provide actionable instructions, and consistently monitored for adherence. Acceptance and rejection criteria. In the analytical world and in the lab world, it must be defined prospectively and applied consistently and not selectively. Data security across all the programs requires enforcing access controls so that every data action is traceable to a responsible individual. And then problem identification. It requires clear, safe, real-time pathway for all the staff involved to raise concerns and have them addressed. Many of these principles are mentioned in our April 24 FDA draft guidance on data integrity for in vivo bioavailability and bioequivalence studies. And I strongly encourage you to review that guidance. So strong quality systems operate in 3 modes simultaneously. Prevent, through SOPs, validated systems and methods, well-defined roles and controls on all data systems. Prevention prevents the probability that problems occur in the first place. Detect, so through active monitoring of audit trails during data collection, independent review points of high risk, data trending and anomaly detection, and internal audits of high-risk workflows. Detection ensures that when problems do occur, they are caught quickly. And then correct, through rigorous root cause analysis, effective and documented corrective and preventative action plans, verification that the plans worked, staff retraining, and meaningful CRO oversight and escalation protocols. Correction ensures that problems don't recur. The goal is not, and importantly, the goal is not a perfect system. It's a system that can identify its own imperfections, respond to them effectively. It's what we're looking for when we inspect a site. And I want to close out with a direct call to action because the stakes here are real. Unreliable or fraudulent data does not just create a regulatory problem. It undermines the safety, efficacy, and quality of medical products that patients depend on every day. It slows the pathway to market for legitimate products, and it erodes public confidence in the regulatory system that's supposed to protect them. To applicants and sponsors, when you select a CRO, that selection is not just a business decision. It's scientific and ethical one. Verify that your CROs have robust quality management systems and demonstrated track record of compliance. You are ultimately responsible for the integrity of the data submitted in your name, even data generated by a third party. And when concerns arise, reach out to the FDA early. The earlier we hear from you, the more options we have to work toward a resolution together. I want to thank you for your time. I want to thank my office. I want to thank OSI, SBIA, and all the incredible public servants that work every day to try to ensure that the public trust is better protected. Thank you for your time. Look forward to your questions.
All right. Thank you for that, Dr. Kassim, for that excellent presentation. We will now open the floor for our Q&A session. As always, please step up to the microphones here in the room. If somebody is speaking, go ahead and line up so we can gauge that engagement here. And in the Q&A chat pod, go ahead and submit your questions now. All right, go ahead.
Dr. Kassim, this is Janak Padia from Primetime Life Sciences. You listed some case studies, examples, some company name. Are those the only company you identify had a problem, or there were many?
Those are the ones that we could put on our public website. So we've had other interactions with folks, but those are just dealt with because they don't rise to the level of
companies.
I'm sorry?
I was wondering how you pick only those companies, one per year.
The ones that rise to the level of documented falsification,
these were the
fabrication.
only company rise to the high level.
Sorry?
These were the only companies they raised to the higher level.
The highest level, yeah.
Interesting.
Thank you for that response. Any other questions in-house this time? If not, please feel free to go ahead and line up at the microphones. We will go ahead and take an online question.
Sure. Although the audit results are public, does OSIS proactively coordinate and share data with CBER and regulatory agencies outside of the U.S.?
Yes. So what's one of the real powerful aspects of the bioresearch monitoring program at the agency is that we do have regular, if not biweekly, monthly interactions with all of the other BIMO centers and sharing information where we have cross of interest of different, the same kind of sites that we're looking at. So CBER, CDRH, CVM, CTP, and the Human Foods Program regularly are able to talk about what's going on. Additionally, we have a significant investment in relations with our foreign regulatory counterparts in the EMA, the European Medicines Association, and the MHRA and Health Canada, which we meet with at least quarterly, if not more often. But we share our findings, they share their findings. We're very much involved in what all the different regulatory authorities have identified and are developing and evolving over time. So that's a really important piece that we're very much interested in maximizing the expertise of everyone as we can across the globe.
Excellent. Thank you for that response. Again, going to prompt in-house. Came all the way out here. Please utilize this time. But if not, we'll take another online question.
Okay. What is the most applicable regulation for laboratories that analyze human serum samples for PK endpoint in clinical trials, as GLP regulation is not directly applicable in such studies?
Exactly, that's right. And I don't think I actually named the regulation, but for the BABE, the bioavailability, bioequivalence, that falls under 21 CFR Part 320, which is a specific subset of the regs that allow us to look at the measurement of bioavailability, bioequivalence in human samples, and defines our requirements for those studies.
All right, thank you. And I think we can probably proceed with online questions unless anybody steps up at this time.
Okay, give me full permission. Here I go. What are the most critical concerns to be aware of when conducting BABE studies overseas?
I think that's an important piece. What we've encountered over the last 10 or so years is a much increased number of studies being conducted overseas. And the things to be concerned with are ensuring that the sites that are used outside of the United States have essentially the culture to ensure that the data that they're generating for a given research program is appropriate, has the ability to identify problems and correct them. Our expectations for foreign data are the same as the ones that we have for domestic data. So it's important that folks recognize that when they're using an outside of the US location, they have to have the oversight and scrutiny of that to make sure that the data being generated is usable.
Excellent, thank you for that. Another online question?
How should identified problems at a site conducting regulated research be reported?
Well, there's a lot of different ways, and I recognize that a lot of times people may see something that they aren't comfortable to report to their management. So we get things in through email. That's definitely the biggest source, I'd say. There are some portals on the web at the FDA site. Some of that is in flux, where we're trying to consolidate ideal ways of collecting information from outside. So, email me. Honestly, just if you can, if you know something, there are ways to get in touch with anybody at the FDA or CDER, and we will find the important pieces of people that need to be talked to make sure that your problem gets attention.
Excellent, thank you for that. Again, another call for in-person questions. We're fortunate we still have some more time. If not, we'll go ahead and take another online question.
Okay. For early clinical pharmacology studies, where do you see sponsors over-controlling low-value processes instead of focusing on true critical to quality factors?
This is a really good point, and I think that it gets into that whole quality by design. We have a unique aspect of particularly of the BABE studies, because they are smaller. They don't require the large contributions of participants that the larger phase 3 or phase 2 and 3 studies do. And so the importance of them being conducted well is critical. We've seen energy being spent on the wrong things. And, instead of making sure that your criteria for analysis, validation analysis, and repeating your samples, which is the core of the argument that your study is supposed to achieve, they'll spend more time on things that are extraneous. And so it's really making sure you're focusing in on what are you trying to accomplish with your study, and that the key pieces of that are well controlled and looked at, and have the appropriate protocols in place to make sure that it can be brought to fruition at the end of the study.
Thank you for that response. We have a few more minutes here, so we'll take another online question.
How will FDA inspectorate share information with other countries to reduce the inspection burden on pharmaceutical and research companies?
So I think that that's similar to what I was saying before, but our relationship with our foreign regulatory counterparts is critical. What they discover on their inspections and what we discover on our inspections is readily shared. When problems are identified, as I said, we meet quarterly at the bare minimum. In real time, if things are identified, we're meeting the next week. We're talking about things immediately so that we can be sure that if we find a signal that we are following up on it and addressing it as quickly as possible.
Thank you for that response. Do we still have more online questions, Brenda?
Oh, yeah, I think I can.
Okay.
I think I can
Then
fit
I'll-
them in.
Okay.
Okay, so let's see.
And again, another call for in-person. I'll keep doing it. Know the drill.
Okay, what is the regulatory requirement for software updates at clinical trial sites?
This is an interesting one because, of course, we've had a unique relationship with electronics and the agency. For those of you who are familiar with Part 11, back in '97, we had a really strong regulation promulgated that established some pretty cutting edge expectations for electronic and computerized systems. But then we sort of backed off on it, and we had a guidance that revealed sort of enforcement discretion. And so that has sort of made it so that all of our different programs have a relationship with electronic enforcement that needs to be improved. It's one of those things that we've wrestled with for years. But for things like software updates, it's important to realize what is that software used for. Control of software changes has got to be under a risk evaluation. What is the software used for? If it's used to collect a critical piece of the study, then you need to be documenting the updates. You need to be documenting what's the changes that those updates have occurred, and that you've tested and you validated that nothing has changed. If it's just something that is used as a calculator, that's not a critical piece of the study, then the level of oversight for those types of updates is not as great. But you really have to make that determination on what's critical to your study.
Thank you for that. I think we have time for one more online question before we adjourn for the lunch break.
Okay. With the FDA's focus on IT advancements and use of technology and AI, do you see a difference now or down the line related to inspections?
I think we are facing a very interesting time, and I think that we will see changes in the future on how things work. One thing that I think is essential, though, regardless of the fancy models that we have to work through vast amounts of data, there will always be a need for some element of boots on the ground, figuring out what happened for the specific information that we're relying on. So I feel like inspections will, of course, change, and hopefully we'll be able to do things faster and more effectively. But I think that there will always be some element that we're going to need to be able to verify in person things that went on.
Wonderful. Well, thank you for that response, as well as all of your information today, Dr. Kassim. We'll give a quick round of applause.
Thank you all so much.
All right, with that being said, it is now 12:10 on my watch, so that means we can adjourn for lunch. We will resume back at 1:10 today. There is Mission Barbecue Catering out back. Just a reminder that the restrooms are around the corner out here. Also, the kiosk will be open with a selection of sandwiches and salads and other items for purchase. And our speakers will be available for any conversations or questions that you might have remaining for those in-person attendees today. Thank you for your time, and we'll see you shortly. All right, and welcome back everybody from the lunch break. We will now begin our afternoon session of day 2 for REdI 2026. Appreciate everybody who's come back to join us so far. Before we get going with our next session presentations here, I do want to give a few quick reminders. If you are here in person, make sure you stop by the registration desk and sign in for day 2 as well. We do use that sign-in sheet to give out the thank you emails with your certificate and evaluation link. So be sure that you have that before you leave today. There will also be another break after these 2 sessions, so you can do it then as well or before you leave campus. Then another reminder for both online and in-person attendees, the recordings of the sessions today will be posted on the event webpage, the live stream recordings, as soon as tomorrow, once we have them available there, so you can expect to see those then. And the thank you email will be sent with the evaluation and certificate of attendance, will be sent to all attendees tomorrow there, so you can keep an eye out for that. Now, with that being said, we will start our next session presentations. Our first one will be called Clinical Trial Innovation, Reducing Regulatory Uncertainty. We will hear from Emily Gebbia, Associate Director of Regulatory Development within the Office of Scientific Investigations and the Office of Compliance here in CDER. Then we will hear from Jessica Bonzo, Lead Pharmacologist in the Division of Pharmacology and Toxicology 2 within the Office of Immunology and Inflammation within the Office of New Drugs, or O&D. At this time, please welcome Emily Gebbia, please.
All right. Good afternoon, everybody. Thank you for coming back from lunch. It's always tough to get the crowd, back on track and keep people's interest when you've had a chance to go outside and get something to eat and maybe some sugar. So I appreciate you being here. And then I also have to tell you that unfortunately, the title of my presentation today is a little bit misleading because explaining how to reduce regulatory uncertainty in 30 minutes is really not possible. And especially because, as I'll talk about, the sources of regulatory uncertainty and how people perceive them are changing all the time, right? So even if I could tell you today how to do it, it would be different tomorrow, probably. So instead, I'm going to try to spend some time today helping folks learn how to embrace uncertainty. I'm going to go for a little bit more TED Talk style than lecture style. Hopefully give you a little bit of helpful information and a little bit of inspiration, because I think that regardless, advancing clinical trial innovation is going to require all of us to accept new ways of doing things despite the uncertainty that goes with that. So here's my overview. Again, I'm going to start out with all the scary things. We'll spend some time talking about those sources of uncertainty and the consequences of uncertainty, but then try to spend some time on how to embrace it, how having the right knowledge, the right tools, and opportunities won't make our uncertainty go away, but can help us figure out how to deal with it. So let's start with this premise that clinical research is complex, right? This is always has been true and always will be true. And that's because we're talking about questions without answers, right? We're not doing clinical research because we already know how things work. So we're inherently in a complex situation. We're also dealing with the health and well-being of real people. Foremost, right, of course, the participants who are volunteering in clinical research, but also the clinical trial workforce. And we're talking about make or break decisions for companies. We're talking about things that really matter to a lot of people. We all have our little spot in this ecosystem. Hopefully you're not the guy up there losing his head. But the point really is that we've already been doing this, right? We're putting in the reps. We've been getting the work done to help us meet a next challenge and lift the heavier weight that we'll refer to as innovation today. Okay. But I do recognize that we are living in maybe particularly uncertain times that add to the complexity of that previous image. Originally, I had wanted to do this as a poll, but I don't really want to call anybody out, making them respond in a room of other people about what their greatest fears are. So maybe I'll just highlight some of the things that I think may be playing in people's minds, and you can sort of internally think about which of these resonates with you. So, of course, we are in a world where we are dealing with geopolitical uncertainty. We're talking about new systems and tools all the time, and by that I mean AI, and AI. In the context specifically of clinical trials, there's a lot of discussion about new approaches, operationally decentralized elements, point of care, pragmatic trials, and different data sources. We have real-world evidence and that sort of thing. Evolution of global standards. ICH E6R3 was published by FDA last year. If you know, you know. And now we're dealing with implementation, and I know that that is a source of a lot of uncertainty. Operational changes and resource constraints. This is at folks' individual companies. Also, maybe FDA faces some of these considerations itself. There are changes in the clinical research workforce in how companies are organized, how people interact in this system, different types of entities and organizations that are popping up all the time. And then we have different U.S. strategic priorities. A lot have been announced at FDA over the past couple of months. This can be another source of uncertainty. So just, we can't do the poll, but just sink in, let all these worries, sit with you for a minute, and think about how these are showing up for you. Another thing that matters, and people have said, that where you stand depends on where you sit, right? So whether you're a sponsor, a CRO, or a vendor, a site, or an investigator, some other clinical research professional, and maybe I can put in here, I know there's a lot of people who are advising clinical research professionals and people, a lot of people in that ecosystem. And of course, patients, caregivers are all going to have different sources of uncertainty and ways that it shows up for them. So sponsors are going to be worried about applications and whether there will be approval or it will be delayed. CROs and vendors are going to be looking at ways that they can expand their services. What can they provide? What's going to be necessary for their customers? Sites and investigators are going to maybe be worried about 483s, hopefully not so much after I finish my talk today. But they also probably primarily want to make sure that they can continue to engage in clinical research and make investigational products available to their patients. Clinical research professionals, I already mentioned some of the uncertainty there, and patients looking for new therapies. And regardless of sort of one of these roles or situations, like we all show up every day as like people with real lives, right? And we care, we have our own things going on. We are worried about our careers, what's going on around us. And so when I talk about regulatory uncertainty, one of the things it's certainly true that it can be a barrier to innovation, but it means something different to every single person. So the key message I really think is that regardless of where you sit and what your thought was on the previous slide is that, you can't just freeze and let somebody else figure this out for you, right? We have to understand that not changing is choosing, and we're all going to need to change the way we operate to meet the clinical research demands of tomorrow and not remain tied to the way things have been done in the past. Of course, that's easier said than done because fear and anxiety are hardwired into our psyche, right? We are cautioned from a young age with many of these sayings up here, it's better to be safe than sorry. So, right, the underlying theme there, action is scary, better to do nothing, right? A bird in the hand is worth two in the bush. I mean, why do we need to catch birds in the first place? Maybe one would be sufficient, you know? And then I think up here we can say, don't go chasing waterfalls. That's mainly for the kids like me who grew up on 90s hip hop. But even TLC was telling us, you know, that we shouldn't be chasing waterfalls. So we get these messages all the time that we should just stick to the status quo. And understanding sort of that predisposition is essential to finding a way to work through it. And hopefully we can do a little bit of that over the next 17 minutes or so. Okay, so embracing uncertainty. We've talked enough about the scaries. Let's talk about how we move forward. I have a quote up here from hometown hero Katie Ledecky, which this is not actually her, so I should be clear. She swims in a pool. She actually has said that she's afraid of the ocean, but the ability to embrace uncertainty leads to ultimate freedom, right? So we need to be able to embrace uncertainty to advance clinical trial innovation. How do we do that? Well, one of the ways is through knowledge. I might not be able to resolve uncertainty, but having information can provide context for decisions that need to be made to move forward. So I'd like to spend my time today on the knowledge section and give you a little bit of information about our good clinical practice inspection process. So what I have up here is the process that CDER uses to inspect clinical trials that are submitted in support of marketing applications. This process starts when a marketing application is received by the center. At that point, my office, OSI, the Office of Scientific Investigations, works with our colleagues in the Office of New Drugs to select sites for inspection. This is risk-based. We have a risk tool that considers 26 different factors across the site and study and application to choose the sites that will be inspected, usually about 3 to 5 per application. The tool we have is a tool, not a rule. It's important to say. So there's a lot of considerations that go into what sites are selected. But once they are, OSI, again, my office, issues an inspection assignment memorandum to the bioresearch monitoring or BIMO investigators in, I have to look now because I still call them ORA, Office of Inspections and Investigations, OII. And those investigators are the ones who actually conduct the inspections, although sometimes OSI will send folks along as subject matter experts as well. Once the inspection is completed, the results are sent back to OSI and are reviewed by the center reviewers. So this process of sending inspection results back to the center is relatively new for certain commodity areas, but it has long been the practice for BIMO. And the reason for that is because the medical and scientific expertise that's necessary to assess inspection findings sit in the centers for BIMO. And that's something I'm going to talk about more in a minute. But after the reviewer completes the review, 2 things happen, okay? One is that a report called a clinical inspection summary is written, and that goes off to the Office of New Drugs to help inform their review of the application. We're going to talk about CISs more later. And then the other thing that happens is that the inspection is evaluated within OSI for a potential compliance action and given a final classification. And these don't sort of happen in serial, they're sort of happening at the same time, but sort of this bifurcation can be important and is something I'll talk about a little bit more. And I wanted to share some inspection-related statistics with you today. So colleagues of mine in OSI published a paper last year looking at GCP inspections that were done in support of a marketing application. So they looked at a 7-year period, and it was just over 2,800 inspections were included in their analysis. And it showed a decline in the number of 483s issued over that time from 24% to 10%. It also showed relatively stable inspection classifications. So there's a slight increase in the number of no action indicated inspections, which basically there are no findings, a slight decrease in the number of voluntary action indicated, where there may be findings, but they aren't of a lot of significance, and then a relatively stable amount of OAIs. Up here it looks like 0. It is not 0 there, but it is less than 1% generally of inspections receive a final classification of OAI. In the paper, they talk about the potential reasons for the decline in the number of 483s, including shifts in GCP compliance at sites, partly perhaps due to advances in technology that make and trial oversight that allow establishments to do a better job of detecting errors and issues in real time and making corrections before they become a problem. But the real reason I wanted to show you this before I have to talk about some other things we're going to talk about is, like, overall, we're doing a really good job, right? I mean, 80% of NAIs and, is great, and decrease in the number of 483s is great. So for the most part, industry, I think, understands GCP inspections and has done a good job of compliance. Yet, I know that 483s loom very large for companies, and so I'm hoping I can debunk a few 483 myths today to take that temperature down a little bit. So I've pulled a quote directly from the 483 itself here, and it says that the 483 lists observations made by FDA during the inspection. They are observations and do not represent final agency determination regarding compliance, right? As I mentioned before for BIMO, the inspection findings are sent to the center for evaluation, and the determination about whether a particular observation is in fact a regulatory violation and whether it's a violation of significance happens at the centers and not during the inspection. And this is because, as I mentioned before, the medical and scientific expertise to assess significance is with the center review staff. They look at things like standard of care, relative risk, physiology, natural disease progression, and a whole host of other things in making that conclusion. So the 483 can get the cards on the table and be a good kickoff for discussions. We always want a response to a 483, but it's not necessarily an indicator of a final classification or of data acceptability. And unfortunately, that's where the flip side of a 483 comes in. So just because something's not on a 483 or a company doesn't get a 483 doesn't mean that everything is good to go. A 483 can still mean, even when there's no 483, there can still be issues for an application. So we have plenty of cases where an OSI reviewer has identified something from their review of the inspection materials that was relevant to the recommendation of data reliability sent to the Office of New Drugs. And it can still also lead to a compliance action without a 483. So next time you're looking in the face of a 483, try to remember it's not the end of the story. It's not the end of the world necessarily, but it doesn't also mean that everything is okay. So we talked about how the clinical inspection summary, or the inspection findings can be used two ways, right? One is for the compliance evaluation and classification, and the other is in that clinical inspection summary. So another paper just published by colleagues of mine takes a closer look at the CISs and regarding the reliability and interpretability of data. So they looked at over 1,200 CISs over 10 years. And I'll note, I said the previous study for 7 years had 2,800 inspections, but now we're only talking about 1,200 CISs in 10 years. And that's because a CIS will cover multiple inspections that are done in support of the same application. So that's why these numbers are a little bit different. And the paper divided up the CISs into three categories. So tier 1 were CISs where there were no GCP concerns. In tier 2, there were GCP concerns, but they didn't substantially impact data quality. And in tier 3, there were GCP concerns that substantially impacted data quality. So here are their findings. The good news is that just under 80% of the CISs found no issues or no significant issues, but 22.5% were categorized as tier 3. And they included an active recommendation, something that the reviewers believe needed to be done before the data could be accepted or relied on for the regulatory decision. And that 22% is obviously a much bigger number than the 10% of inspections that receive a 483, and a much larger number than the less than 1% of inspections that are classified as OAI. And the reason is that a lot of the issues that are driving these active recommendations are from NAI and VAI inspections. They're because of variations that we see across clinical sites, because we're doing more than one, or they're things that aren't necessarily regulatory violations that drive those classifications, but that still can impact data reliability and interpretability. So we're going to talk about quality in a little bit, but the reason we do so much talk about quality is because when you're making decisions based on the fear of findings in a 483 and instead of about what's best for the trial, we may not be getting the results that we want. You might avoid the 483, but you might not be out of the woods in terms of the acceptability of the data for review. I already made that point, but there's the actual bullet that says it. Okay, so let me talk a little bit more about what these active recommendations look like. Most of them were either requests for additional information from a sponsor or the conduct of an additional statistic analysis, like a sensitivity analysis, or to include updated data. There are a few, though, that had more significant actions or recommendations, including not including data from certain sites or certain parts of data, and that can obviously change the ultimate analysis when that data are excluded. Requesting audits either by the sponsor or by a third party, which can lead to delays in the ability to meet that timeline that everybody's driving towards, as can the need for additional inspections. And in very rare cases, there can even be need for a new trial altogether. So again, this is to emphasize why turning down the temperature on 483s, is maybe that's not where the focus is best placed, right? And by focusing on the quality, the things that are really going to matter for the application, instead of just trying to get through an inspection, might be leading to better results for folks. So that's the information I have to share. Now I want to talk a little bit about some tools for embracing uncertainty. All of these are things that actually have been talked about at other parts of this conference. So again, my goal here today is not to provide you all the information that you need in now 10 minutes to embrace uncertainty, but to help point you in the right direction and help you understand how some of these things fit together into a bigger picture. So you will hear anybody probably that speaks from OSI talk about fit for purpose clinical trial quality. Regardless of whatever uncertainty is out there, our goal should always be the same, which is to conduct quality clinical trials. Fit for purpose clinical trial quality is quality that's sufficient to meet the trial objectives, provide confidence in the results, support sound decision-making, and protect participants' rights, safety, and welfare. Fit for purpose clinical trial quality acknowledges that trials are rarely conducted perfectly. Striving for perfection is not realistic and it is not necessary. We know that there are going to be errors in a trial. What we want to do is to avoid errors that matter and that could have an impact on the overall trial results. So you might be asking, well, how do I do that? Well, here you go. This is how you do that, hopefully. The first step is to make sure you get the fundamentals down. Quality by design is not an innovation, but it is the cornerstone to making sure that any innovation you are using in a trial has the best possible chance of succeeding, because quality by design is building in quality from the start of a trial. The success or the likelihood that you'll have a fit for purpose clinical trial quality can be improved through prospective attention to preventing important errors. And you might be thinking, I thought you just said that you know that errors are going to happen. Well, that's true. They will happen. QBD is about preventing important errors, but also there are errors that we won't be able to prevent, right? And so the goal is to mitigate those errors to the extent that we can through risk-based quality management. This is not about doing less, it's about doing better. So our risk-based quality management is not about doing less, it's about doing better. Both QBD and RBQM, just to use as many acronyms as I possibly can in 30 minutes, have at their core focus critical to quality factors of a trial. So these are the data, systems, and processes that it's crucial to get right to ensure the reliability of results and the right safety and well-being of participants. A few resources. Luckily, during this conference, you heard from one of the leading experts on QBD and RBQM, my colleague Cheryl Grandinetti. She spoke yesterday, and I recommend her talk to you. As I mentioned before, the ICH guideline E6R3 has been published, and the ICH team, in conjunction with MCRT, has published a training that's available online. A few of the modules are already available, and others are forthcoming. I'll talk about C3TI in a minute. All our guidances are online. And then I just really want to commend to you an upcoming workshop between FDA, MHRA, and Health Canada. That's the first week of June. You can still register, I believe, for virtual participation. And if you look at the first day's lineup, it's just like QBD and RBQM all the way down. I mean, the slates of speakers and the topics they're going to be covering are great for people who want to learn more about this, and I really hope that you do. Okay, next, I felt like I couldn't not talk about artificial intelligence, but also I probably should not be talking about artificial intelligence since my most successful use is coming up with puns to embarrass my children. But obviously, this is a tool that FDA is interested in. I know all of you are interested in. Annie gave a talk yesterday all about AI, and so I would recommend that to you. Just a couple things specific, more related to clinical trial innovation. This joint workshop that was held in 2024 is available online. That was held with the Clinical Trials Transformation Initiative, and would be interesting listening for folks. And also a paper that was co-written by folks from my office, foreign regulatory counterparts in industry. And I just want to make a brief aside and say this is now the third article that I've mentioned that has been published by somebody in my office. And I just think journal articles are a really good way for us to communicate information that's not guidance, which necessarily has to sort of be future-proof and forward-looking, or compliance actions, right, which are talking about things that have happened in the past. These journal articles tend to be a little bit more of a snapshot in time. This one in particular talks specifically about artificial intelligence in clinical trials and has a lot of really great information. So I will recommend that to you. And looking at my time, I will just keep on going. So finally, the CDER, Center for Clinical Trial Innovation, or because we love our acronyms, C3TI. My colleague Meghana Cheslani did a whole talk on this yesterday, and I'm sure that there's a ton of great information there. But overall, the goal of C3TI is to improve the efficiency of drug development through innovative clinical trial approaches. The program helps to facilitate collaboration across already existing and new innovation programs in CDER, provides a ton of information, including that knowledge compass that I showed earlier on different innovations. And then there's opportunities for engagement. I've participated in some of these engagements. They're really informative and are bringing a lot to light in terms of what's possible for clinical trial innovation and how to get over some of the perceived and real barriers that there are. Okay, all right. So now here's where we're going to get to real TED Talky. We've talked about some of the sources of uncertainty, how to embrace them through knowledge and tools, but how do you find opportunity in uncertainty? One of the reasons, as my boss likes to say, that the windshield of a car is so much bigger than the rear view mirror is because what's most important is what is ahead of you instead of what's behind you. So while your rear view and side view mirrors can provide you context, we really need to be focused on what's coming next and how we can navigate that. So how do we do that? How do we turn knowledge and tools into action? We do need to acknowledge reality, right? Hiding from risks and uncertainty is not going to make them go away, however much we'd like that to be the case. And we talked about how things are always changing, so it's important to maintain situational awareness at all times. You're here, which probably means you already know this, and I appreciate it. We need to keep the focus on what's important, not 483s, in my opinion. And we don't have to run the whole race at once, right? Step by step, we can make little changes. As we like to say, what's the next right thing? Well, actually, Anna says that in Frozen, but I've incorporated it into my talk. And then, being flexible because things are going to change as we go. So just my final point here, one of the biggest impediments we've seen to the uptake of clinical trial innovation is changing cultural mindsets. It's not any of the uncertainty. It's not any of the regulatory barriers. It's not any of these other things. It is getting people comfortable with a new reality. And so I hope today I gave you a little bit of knowledge, some information about some tools that are available to help you embrace uncertainty and move forward boldly to adopt innovations that hopefully will help lead to new cures faster for patients. That's all I got. Thank you guys so much. I really appreciate you being here.
Presentation. Thank you for that, Emily. So in the name of embracing no uncertainty here, you guys already know what's next. If you are in the room, please line up at the microphones as we begin our Q&A session. We will begin with our in-person questions. If you are at home, please submit your questions in the Q&A chat pod at this time. We do have a question at the microphone, so we'll start here.
Yeah, thanks, Emily, for the great presentation. I really enjoyed all the references that you made it very exciting. My question is expanding upon maybe your topic of AI. I know it's not a forte of yours, but if you could help us understand maybe in your office or across FDA for inspections, how is AI being used, and to think of a couple of recent examples that may or may not still exist, like the one-day inspection pilot and the real-time clinical trials initiatives.
Yeah, thanks for the question. Not surprised to get an AI question. I can't really speak to the real-time clinical trial pilot or the one-day inspections or uses of AI in those. I will say there's a lot of curiosity about AI and how it can be used. There's a lot of experimentation. There's a lot of folks who, I guess government agencies, regulatory agencies can sort of maybe get a reputation of being a little bit Luddite, and slow for the uptake. But I have seen people who are just really excited about the possibilities and really curious and experimenting and engaging across the office and the center and the agency to learn from each other and see what possibilities are out there. So I think we're really in early stages of trying to see how things would work, like people like me just learning the tools and getting comfortable with it, and people who are really out there on cutting edge sort of stuff. So I expect that there will be a lot more and just stay tuned. Although, I still think there's, Sean mentioned this earlier during his talk, inspections, there's still going to be the need for humans in the loop and boots on the ground, and the same thing with our reviews, but ways that we can make our work more efficient. And I think folks are really open to that and are actively working on figuring out how we can do it.
Excellent. Thank you for that response. Again, if there's any other in-person questions, please line up at the microphones at this time. I'll go ahead and let people, if there's anything else, looking around, any in-person? Okay, we do have one in-person question.
I have a question about the risk management or the risk-based approach. What types of documentation or what would you recommend as a starting point for documenting risk assessment, whether you're at a sponsor or a site? If that's the direction that we're supposed to be going, do you have any thoughts on how to get started with documenting your risk assessment?
Yeah, excellent question. I think this is something that I think we know that there's interest in getting a little bit more specificity out on. But the number one thing is do document it. We go in plenty of times and we hear retrospectively go, we thought about this, but it's not documented. I think there's sort of a recognition that, an expectation that there is some sort of documentation that shows the thinking, right? Like show your work. This is what we identified as our critical to quality factors. Here's what we identified as our risks. Here's what we evaluated them. Here's what we did. And then that should not be static, right? The big part of risk, quality risk management is doing evaluation over time. So is it a regular check? And, there's what's an ICH E6R3, but it's still at a pretty high level. And I think there's a lot of room for companies to right size their approach based on what it is that they're doing. And so I think that the expectation is sort of that the level of detail would dovetail with that. But as much as you can prospective, like saying these are the things that we considered, and then when things come up, say this is what we identified, how we identified it, what changes we made. It just makes it so much easier when we're going in and there's a problem. Say, yep, we know, we thought about this. Here's how we were prepared. Here's how it came up anyway. This is what we did in response. This is how we managed it on an ongoing basis. You can't sort of, it's much easier to have that done already because you were monitoring as you went than have it created for purposes of the inspection. But I can appreciate that, a part of the challenge of doing it is it does look so different. There's so much variability in what may be needed for a particular trial, but that there is interest in having some more specificity that would be helpful to sponsors and sites and other organizations and what that might look like. There's no wrong answer, I guess, is the overall, as long as it's right-sized for the trial or other activity that you're engaged in.
Excellent. Thank you for that response. Again, if there's any in-person questions, line up at the microphone. We will take one online question at this time.
Okay.
We're going to take one online one. But you can remain at the microphone if you'd like.
Despite strong advocacy for QBD, its implementation often remains superficial in industry. What regulatory incentives or expectations could drive deeper, more authentic adoption?
That's a very good question. I think it's important to recognize that although quality risk management itself as a concept isn't new, sort of the articulation of it in E6R3 is new. It's baby steps. It's baby steps for everybody. I don't want to sort of, this is a mind shift for FDA as well in terms of how we're evaluating things and looking at things. So one of the hopefully takeaways people got from my talk is like, we don't have to run the whole race right now. We have to figure out the best next thing. And so while implementation may seem superficial, sometimes you have to start somewhere, right? And the hope is that as industry, as FDA gains experience with this, that we will be able to start to develop it and be able to share more best practices, check out those C3TI demonstration programs. We'd love to have people participate to help generate this knowledge base. But, it may seem superficial, but I see it as a good first step to getting to what's really needed.
Wonderful. Thank you for that response. And it looks like we have time for one more question. So we'll go to our in-person question now.
Thank you for the great presentation. Very informative. My question is regarding the DHT. I know the agency probably published the guidance 3 years ago. Want to see can you share some insight and observation? What do you see the trend? And also what's typical common gaps during either the drug review or during the inspections.
And so you said the guidance on, for what was the guidance on?
Digital health.
Digital health technologies. Yes, yes, yes. Good question. This is where I would usually use phone a friend. No, I think it's really exciting. I think that digital health technology is sort of, I think, an innovation we're really interested in because it really gives us the opportunity to bring trials to participants where they are. We do see issues in inspection related to data capture and that sort of thing. And so this is, again, where quality by design and RBQM are really important, thinking about like what data are you using the digital health technology to generate? What are the risks? Are you giving, is it something where you're going to have complicated passwords and that people are going to forget? And, really thinking through how that technology is going to be used, what it's going to be used for, and prospectively planning ways that you can mitigate some of the risks that are associated with them instead of trying to troubleshoot everything on the back end.
Excellent. Well, thank you for that and also for the informative presentation. Looks like that's all the time we have questions for today. But thank you again, Emily, for your presentation. If you'll join me in thanking her. And at this time, I will now welcome Jessica Bonzo for a presentation on streamlining non-clinical safety assessments. Thank you. Okay. So this topic is pretty broad, so hopefully I can give you a few useful nuggets in the next 30 minutes. As I said, I'm a lead pharmacologist within the Division of Pharmacology Toxicology for Immunology and Inflammation within the Office of New Drugs in CDER. There we go. So it has been 1 year since the FDA published the roadmap for reducing animal testing in preclinical safety studies. And we've had many notable accomplishments over the past year. And just to highlight a few of these accomplishments includes the ISTAM program, which was made a permanent program for the qualification of drug development tools. We've also published several publications on NAMs, as well as we have our new draft guidances for monoclonal antibodies and the NAMs validation guidance. So in today's talk, there's a lot in this to talk about. We have the S1B addendum to the S1B testing for carcinogenicity of small molecules in pharmaceuticals. And then we'll follow this with talking about some notes on our new draft guidance for monoclonal antibodies, the approach of NAMs within CDER, and finally, a few of our ongoing and future initiatives. So I would be remiss if I didn't first mention our experiences in oncology, because my talk today will focus more on non-oncology indications. While ICH M3 and S6 apply broadly to all indications, ICH S9 was specifically developed to guide the non-clinical evaluation of anti-cancer pharmaceuticals to provide effective therapies more expeditiously. And subsequent publications by our colleagues in oncology have provided even further flexibilities than what was initially proposed in S9. And the principles in the new monoclonal antibody guidances and the NAM guidance can be used to support additional streamlining in oncology. With that said, I'm going to focus more on non-oncology chronic indications. And this is just a snapshot of typical NME programs that you might find for a small molecule or a biologic. And all these studies may not necessarily be conducted. It's just an array of what could possibly be conducted. And today's talk, I'm going to focus on animal studies for carcinogenicity assessments for small molecules, and the general toxicity assessments for biologics, and then also NAMs, which we might classify as special studies, but they could also be incorporated in any of these areas on this slide. So the S1B guidance. This is an example of a science-driven process that was rapidly adopted by regulatory agencies that is producing tangible results in reduction of animal usage today. The addendum is to be used in conjunction with ICH S1A, S1B, and S1C guidances, and describes the carcinogenicity assessment for small molecules.
The addendum is an integrative approach that provides specific weight of evidence criteria whether a 2-year rat study is likely to add value to a human carcinogenicity risk assessment. The addendum does also explain an exposure-based dose setting for transgenic mouse studies, but I will not be discussing that today. So this is a stepwise process. First, the sponsor will gather all the data about their drug product. This data may be internal proprietary data, or it may be sourced from published literature. Then the sponsor will compose their integrated analysis of weight of assessment factors. These factors include the target biology, results from your secondary pharmacology, which would be off-target analysis, histopathology from your chronic tox studies in your potentially 2 species, hormonal effects, also your results of your gene tox battery, and finally immune modulation factors. So once we have this analysis completed, there's 3 outcomes. The first outcome could be that carcinogenic potential in humans is likely. If this is the case, a 2-year rat study would likely not be needed and would not be warranted to add the information, and possibly even a mouse study would not be needed. Second, the carcinogenic potential in humans is unlikely. In this case, also a 2-year rat study would not be warranted. However, a mouse study could possibly still be warranted. And finally, the human carcinogenic potential is uncertain. Therefore, a 2-year rat study would add value. And a mouse study may also be warranted. And the most frequent reason for uncertainty is usually around the target biology, especially if the drug is first in class. And we encourage you to look at all sides of the published literature. Our reviewers do have access to PubMed, and we can review all the background literature on your target biology. So just presenting one side of the argument usually won't cut it. So this is an example of a case study of what we might typically see for an S1B case study, and I've presented it with each of the different weight of evidence factors that we look at. In this case study, we have a small molecule that's a regulator of cholesterol metabolism. There are multiple approved products in this class, and human loss of function mutations show no increased risk. And there is conflicting literature on the role of the target in tumorigenesis. Secondary pharm screens were pretty clear. There was no concerning binding. And histopathology from the chronic tox studies in a rat and a dog did not have any concerning findings related to hypertrophy, proliferation, or pre-neoplasm. And there were no effects on hormonal effects or immunomodulation, and the gene tox battery was negative. So if you were presented with this weight of evidence, what would you decide would be the appropriate approach for the carcinogenicity assessment? The sponsor decided that it was unlikely to be carcinogenic in humans and that a 2-year rat study would not add value. They did propose a 6-month transgenic mouse CARC study, and this is evaluated through a different process through an SPA analysis. And so the division agreed that the human CARC potential was unlikely, and therefore that the 2-year rat study would not add value. The division presents this analysis to the ECAC, which is the Executive Committee for Carcinogenicity, and the ECAC concurred with the division's recommendation, and these recommendations are then communicated to the sponsor that a 2-year rat study would not be warranted. So this first topic, we have, we've had approximately 60 weight of evidence submissions now in this process, or in almost 3.5 years. And about 60% of these submissions has been determined that a 2-year rat CARC study would not add value. And so we just do a little back of the napkin math here, and we've estimated that approximately 25,000 rats were probably not used for regulatory 2-year rat studies. So this has been the impact of this program, and it's been a resounding success in our opinion. So the next guidance I'm going to talk about is our new draft monoclonal antibody guidance that was released in December. And so the scope of the guidance, and this is a pretty new one for us now. It is specifically for monospecific antibodies. It may apply to multispecific antibodies or antibody drug conjugates only in certain cases. So we're really just focusing on monospecific right now, but many of the principles could apply. And we believe this guidance will be most impactful for chronic non-oncology indications, since oncology already has existing flexibilities in their non-clinical program. So how are you going to evaluate the safety and support of chronic administration of your monoclonal antibody? Your non-clinical toxicology program may be conducted solely in a rodent species if it is determined that the monoclonal antibody is similarly pharmacologically active in humans and a rodent. And second, studies of 3 months duration plus a weight of evidence to support the chronic administration is expected. I'll caveat this statement with saying that agreement from the review division should be sought for this approach. And there may be cases in which the conduct of a toxicology study more than 3 months is warranted, as when the pharmacodynamic effect of the monoclonal antibody doesn't initiate until, say, 2 or 3 months after administration. So what are the weight of evidence risk assessments for monoclonal antibody factors that you should think about? So we have mechanism of action and the primary pharmacology of the drug, the toxicology and PK data from the non-clinical program, knowledge of secondary pharmacology effects, if there are any, clinical safety and PK data for chronic administration that may be available from non-US approvals, information on toxicities associated with the target from other approved products or published literature. And finally, if there's existing information from transgenic animals, surrogates, NAMs, those also could be used. But I will say there is no expectation that you conduct these studies with a surrogate or the transgenic just to support the weight of evidence. You may have conducted them for other purposes, though, in your program, and they could be useful in this assessment. So, but there are also times when even a 3-month study would not even be warranted. In these cases, such as when an antibody is directed at a foreign target, if the antibody does not bind to the target in a commonly used animal species or does not elicit the pharmacological effect, or if there is substantial animal data against the same molecular target that indicates that the animal data has not been predictive of the human toxicity. These first 3 examples are scenarios where no animal studies might be warranted and the toxicological risk assessment is based on in vitro and weight of evidence. The final 3 examples, such as initial toxicity studies, indicate the formation of neutralizing or clearing ADAs, such as there's little to no drug exposure, there's exaggerated pharmacodynamic effect, severe immunosuppression as one example, or perhaps the existing pharmacology data and experience with an established class of drug is extensive, studies of 3 months or greater duration may not be warranted. And then the final section of this guidance that I'm going to elaborate on today is the DART assessment. You should begin with a thorough weight of evidence assessment using principles that have been outlined in this draft guidance. The ICH S5 guidance for developmental and reproductive toxicology would also be a really good source to look at for weight of evidence factors. If there is no known DART risk, no further studies would be warranted for the monoclonal antibody. Similarly, if it's unlikely to pose a developmental or reproductive toxicity risk, no further studies would be warranted. The final category is the uncertainty one. And at this point, we would recommend that you explore NAMs, lower phylogenetic species, or alternative approaches, and seek alignment with your review division if you would want to pursue one of these alternative approaches. And hopefully through the use of these alternative approaches plus weight of evidence, the risk will be sufficiently characterized and no further studies would be warranted. As a last resort, if there is still substantial residual uncertainty, an EPPND study in a sexually mature NHP may be warranted. But again, seek guidance from the review division and concurrence that an EPPND study would be needed. This guidance is fairly new, and we're already seeing great interest in the flexibilities that are outlined for monospecific antibodies. Hopefully, We'll have a little bit more concrete numbers to share soon, but in lieu of that, we've provided some rough estimates on what we think may be the potential impact this guidance will have on the use of NHPs in regulatory toxicology. Again, these are very just rough estimates of what you could expect to animal numbers in a NHP chronic toxicology study, the number of sexually mature female monkeys that might be used in an EPPND study. And when we do this, combined with the number of studies that we have typically seen over the past few years that are submitted each year to OND, we estimate that upwards of 1,700 adult monkeys would not be needed for regulatory toxicology submissions per year. Okay. So now we're going to move on to an even bigger, broader topic that can actually affect animal usage in both the small molecule and the biologics world, NAMs. So we're encouraging the use and submission of NAMs for regulatory purposes is a key part of the FDA's roadmap, and CDER released the draft guidance on NAM validation 2 months ago. And I believe the public comment period is closing soon, and we'll have finalized guidances coming in the next few months. And what are NAMs? I think the definition changes every day. I think everyone disagrees every day what a NAM is. Is it a weight of evidence plus a technology? Is it just a technology? So this is the definition that we're going to use today and that we have used since 2020 within the FDA. But don't take my word for it. We could change it tomorrow. So the new approach methodologies include a broad range of methods, such as in vitro, in chemico, and in silico methods. In vivo methods can also be considered NAMs when they improve predictivity, shift studies to phylogenetically lower animals, or otherwise help replace, reduce, and refine animal usage in development programs. And that statement was actually published in the first of a series of papers that we have released out of CDER on our perspectives on the use of NAMs. These papers have also covered topics such as the gaps, knowledge gaps in non-clinical studies that NAMs would be, that are desperately needed to fill. And we also have some quantitative analyses of what NAMs we have received within OND. Let's see here. So let's talk a little bit about what we have received within OND. This paper was just published about a month ago, I think it was now, out of Nikisa Sadrea from OND Senior Leadership. So the predominant type of NAM that we have received within OND are in silico, at 49% of the submissions being such technologies as QSAR or read across. And the second largest category that we have are in vitro studies. And these are cell culture studies that a lot of people talk about, such as microphysiological systems, organ-on-a-chips, co-cultures, et cetera. And this in vitro category is the most diverse bucket in terms of technologies. And we receive more submissions. This analysis can be further refined to identify which types of in vitro technologies are really being used the most and the most informative for regulatory purposes. And we're hoping with the publication of our guidance, these numbers will increase significantly very soon. So to the nuts and bolts of this guidance, we have okay, let's see here. Oh, sorry. Okay, so CDER defines validation process of NAMs as accuracy, reliability, and relevance of a procedure are established for a specific context of use. The guidance does not address qualification. Qualification is a determination that a drug development tool and its proposed context of use can be relied upon to have a specific interpretation. The Ames assay would be a classic example of a qualified NAM that can be used across multiple drug development programs. And the iSTAND program at FDA is our current drug development tool qualification program. I'm not going to talk about that because that's another whole day of discussion that we don't have time for. So the features of the validation that CDER has published. And this publication is very similar to some of the ICVAM pillars that were published in 2024, but they've been put in the context of use for drug development. So first is context of use. Clearly define the intended use of the regulatory purpose of your NAM. human biological relevance. Describe the features of the NAM and how they relate to human biology. Technical characterization. This ensures that your NAM is robust and reliable and reproducible. And then fit for purpose. A NAM is fit for purpose if it assists CDER in regulatory decision making. Additional considerations are the FDA does not require a NAM submitted in a drug application to undergo qualification through ISTAND. A NAM can be submitted in an IND, and NAMs should be fit for purpose and used in the context of your weight of evidence. Oh, am I over time? I lost my timer, but okay, we're good. We're good? Okay, sorry about that. So NAMs in non-clinical safety assessment. We've actually been using these for decades. They just weren't called a NAM. They were just technology and they were good science. So we've been viewing NAMs in different areas, including ocular toxicity, phototoxicity, skin sensitization, other local toxicities for decades. So originally, say for ocular toxicity, we would have in vivo assays such as the rabbit eye irritation or Dray's test. And science evolved, and we no longer need to use animals for this in vivo test anymore. We have a validated in vitro and ex vivo NAMs that can use human corneal-like epithelial cells and model irritation in the eye. And there's even guidances through OECD with multiple different technologies that you could possibly use. So we also have another example of where NAMs could refine your non-clinical program. For biologics, off-target effects are frequently measured using the tissue cross-reactivity assay. The TCR tells you where a biologic might bind, but it doesn't tell you what. And so now we have cell-based protein arrays that can be used to tell you where what is being bound, and then you can combine that with the where to help inform your weight of evidence of possible risks associated with this biologic. So that was a really fast whirlwind through some of our programs for this past year. A lot is happening in our space, and we even have more coming out. One of the first areas that some of you may be aware of is that the ICH S13 guidance on monoclonal antibodies is currently under development, and the oligos sit in a unique space between small molecules and biologics, and there's hope that there's opportunities to refine and use NAMs for some of the safety assessments in oligonucleotides. Severely debilitating and life-threatening indications, SDLTs. And some of you who know our guidances by heart know that we have an SDTL guidance for non-malignant hematological indications. But there's a lot of us out there that don't work in hematology, and we still have SDLT indications. So there is an active effort underway right now to develop guidance that will incorporate the principles that were developed under the hematologic guidance and to expand it through all different indications. And lastly, rare diseases are a huge priority for FDA to provide therapies and non-clinical assessments. And these, we've always had flexibilities, but now we're working on guidances to incorporate that into other guidances to really clearly express to sponsors where your flexibilities opportunities could be in rare diseases. So there's our whirlwind. So I hope you see today that we have successful implementation of ICH S1B addendum. This is an example of a science-driven global regulatory authorities coming together to reduce the usage of animals. We're having rapid uptake in the recommendations outlined in the draft monoclonal antibody guidance. And also, reviewers are ready for NAM submissions to support safety assessment. We've had thorough training. Many of us have also worked in the NAM spaces before coming to the FDA, and we're excited to see where the science is and how to better identify risks and make drugs safer for humans. So thank you very much to the SBI organizers for inviting me. Thank you to the OND Senior Farm Tox leadership who has helped with many of these guidance updates, and in particular to the Division of Pharmacology and Toxicology for Immunology and Inflammation that helped together to put together this presentation. And when in doubt, ask. It's always best to submit an interact meeting early in your development phase, or submit a pre-IND meeting, or submit a type D meeting once you're already like in development, to ask us about these, especially the weight of evidence of when is it appropriate to use an animal species, or when is it appropriate to use the weight of evidence or the NAM. So we're always here to answer your questions.
All right, thank you for that excellent presentation, Dr. Bonzo. We will now open up the floor for any questions. Again, in person, there's the 2 microphones, to stand up at them. And if I invite everybody that has questions to stand up now, you can also stand up and stay at the mic as well. Are there any in-person questions for us today? Okay, looks like we do have one. If you are online, you can submit those questions now as well. Go ahead.
That was a great talk. I really liked the overview of the accelerating non-clinical safety studies, monoclonal antibody guidance. When I read that draft guidance, I definitely had a different perspective on what the section on when doing a 3-month or longer safety study is no longer necessary. And I think that really speaks to maybe some of the confusion in the space right now. There's a lot of changes, and I like that you mentioned interact meetings. But is there any upcoming training opportunities aside from this one, obviously, which is great, but specifically for reviewers and pharmaceutical companies and sponsors to come together and to learn more about what would constitute sufficient weight of evidence to avoid those studies? Thank you.
Okay, thank you. That was a great question. So if those weren't able to hear it, the question is about learning opportunities for both sponsors and as reviewers to really understand the weight of evidence concepts in the monoclonal antibody guidance. So the guidance it was open for public comment. It is being actively worked on as we speak, and I know the final guidance will be coming soon. So hopefully that, first of all, the update on the guidance, I hope will clarify some of the content that was originally published, and that's the point of the public commenting period. Is maybe to us it made perfect sense, but I guess it didn't make perfect sense to everybody else. So hopefully we'll have that update coming soon. There are many speaking opportunities, especially from our senior leadership, that will be upcoming. There are so many requests these days. I couldn't even tell you how many lists. You just keep looking for us out there. We are coming. I know we had Dr. Sadria spoke, I think was yesterday on NAMs. I know we have leadership speaking tomorrow on different ways to right-size non-clinical programs at a different conference. So we're definitely out there. We're trying to get the message out there. I personally don't know of any specific trainings, but we'll definitely be out there talking about our perceptions and helping sponsors understand the new guidance.
Excellent. Thank you for that response. Again, are there any other questions in-house? Okay, if there are no other questions in-house, as always, please feel free to line up to the microphone. If you do have a question at this time, we'll jump over to the online questions.
First online question. We are mainly working on bispecific antibodies now. Can the MAP guideline be extended to bispecifics?
Okay, so the question is about the bispecific antibodies or multi-specific targeting antibodies. And yes, you can use some of the concepts that are in the monoclonal antibody guidance to apply for the bispecifics. We've actually been doing this without a guidance for several years. We always take every IND that we receive as an individual case, and we look at the evidence presented on whether or not different animal studies are warranted in each case.
Excellent. Thank you for that. Are there any other in-house questions? If not, we'll take another online question at this time.
Are there any ongoing joint efforts between the FDA and other regulatory agencies to streamline the requirement of non-clinical safety studies in the reduction of animal usage to support drug development?
Okay, so the question is about if there's any kind of international group organizations working on this to harmonize across the globe. I assume there are. We have ICH. I personally am not at that level. I don't work on those. But yes, it's anytime we speak with any of our colleagues across the world, everyone is very interested in streamlining and reducing animal usage and making the best scientific decisions possible.
Thank you for that. Let's go take another online question at this time. Or is there another in-room question? Looks like we do have one in the room. Go ahead.
I'm sorry, I definitely talked too much. So that actually brings up another question that was in the roadmap, and specifically, I don't think you mentioned this in the weight of evidence approach, but the opportunities to incorporate existing clinical trial data for drugs from other regulatory agencies, and whether or not that can substitute for animal testing. Does that did I explain that well?
So your question is using essentially foreign
Yes.
clinical trial data to substitute
Yes.
non-clinical. I don't think I would use the substitute word. It is an area that's evolving, that involves many moving pieces because it's not just non-clinical that is involved in evaluating the utility of foreign clinical data. So that would be a decision that involved our clinical teams and possibly our stats teams, the clinical pharmacology teams. So it's not an easy yes. We can use it. It's really an integrative approach, and sometimes it may be appropriate, and other times it may not be.
Excellent. Thank you for that. Thank you for your questions in-house. You can continue to go ahead and line up at the microphones in-house if you do have questions. Luckily, we have more time for some online questions at this moment.
How do we get a review division's feedback on whether a NAM will be accepted?
So you can submit for just your own particular IND. You can submit the NAM and the study report to the IND in the form of a meeting request. This would be a great opportunity to use the Interact format. And we request that you put as much detail and data about your NAM and your approach and the context of use that you're intending to use it within the study report. And within the package so that we have the ability to review everything and see how it might fit into the non-clinical program.
Thank you for that. We can take another online question at this moment.
How long does it take to get an answer regarding my S1B weight of evidence submission?
Okay, the length of time for an S1B answer. So S1B is not currently under a PDUFA deadline. We do have our internal goal dates, which would be 3 to 5 months. Sometimes we can get them out earlier, and sometimes it does take a little bit longer.
Thank you. Again, another reminder for the in-person questions. If not, we'll continue on with the online questions since we have ample time.
Does FDA accept chronic studies in lieu of carcinogenicity studies?
Does we accept chronic studies in lieu of a carcinogenicity study? The chronic studies conducted in the rat would be needed to do your full weight of evidence assessment for the S1B process. So it's not in lieu of, but it is a component of your S1B analysis.
Thank you for that. Are there more online questions, Brenda?
Yeah, we have a couple more we can go through. Can transgenic mice be used instead of NHP?
So the question is, can transgenic mice be used instead of the NHP? So we've said you can use a rodent as your non-clinical species. You would need to provide adequate justification that the transgenic animal is an appropriate model. And so I would seek guidance from your division and to see if they concur with your approach.
Excellent, thank you. All right, more questions online?
Yeah, I think I have a couple here we can do. Can I apply the concepts in the monoclonal antibody guidance to bispecific antibodies, even though they are out of scope?
Well, I think we've answered that one again, but yes, You can apply the concepts from the monoclonal antibody guidance, which is more intended for a monospecific antibody. You can apply it to tri-specific antibodies as well.
Okay, let me just get myself right over here.
All right, one more reminder for in-person questions. If anybody has any ones, we have time for a few more questions, potentially, if there's more online or in person. You know the drill at this point with the lining up, but if not, I'll shift back over to Brenda for the online questions.
Okay, what is expected for a weight of evidence assessment for DOT for MAPS?
What is expected for a weight of evidence assessment for DART for monoclonal antibodies? So again, you look at all different factors that would be laid out in the guidance. Many of the factors that you would consider for both the chronic monkey study would be applicable for determining if you would need to conduct a DART study. And also the ICHS 5, which is the Developmental and Reproductive Toxicology Guidance, that also has many factors on weight of evidence of whether or not certain factors would be important in deciding the risk to humans. So you can use both of those resources.
Excellent, thank you for that. All right. So let's go to another online question at this time.
I would have to say we have exhausted our online questions.
That is great news.
I did finish a little early.
Yes.
Sorry
Last
about that.
call. We do have a little bit of time, so last call for any in-person questions, anything burning. Okay, while I've got you here though, if you are in person and you were a walk-in today, make sure that you also leave your email address out front. So before I let you go for the break, if you haven't done that, make sure you have that so you can get the thank you and the survey and evaluation link tomorrow. We just don't organically know that off the top of our heads. So you'll have to tell us either during this break or before you leave today. And with that, we can conclude for our 15-minute break. We should resume around 2:45 for our final session of the day, and you're good to go now. All right, so it is 2:45, so I think we are good to begin our session now. Welcome back, everybody from the break. I hope you got to stretch your legs a little bit, get ready for our afternoon session that will be update on rare diseases at FDA Center for Drug Evaluation and Research from Dr. Scott Winiecki, Acting Associate Director for Rare Disease on the Rare Disease team within the Division of Rare Diseases and Medical Genetics. That's within the Office of Rare Diseases, Pediatrics, Urological, and Reproductive Medicine, and the Office of New Drugs. This time, if you will welcome me in joining Dr. Winiecki.
Thank you, and just thank you to everybody in the room who's still here and came back from the break. I appreciate it. It's always difficult being the last talk on the second day of a conference, and appreciate all those online who are still hanging with us. So I hope that you will have spent your time in a valuable and worthwhile way in the next half hour. So first, I want to just talk about where I work and how I'm found on the org chart. So if you look, we're in the very long titled office up there. So if you see the Office of New Drugs and you move all the way over to the right, we are the Office of Rare Diseases, Pediatrics, Urologic, and Reproductive Medicine. So that encompasses a vast amount of things. And then I am right here, the Division of Rare Diseases and Medical Genetics, or DRDMG. So if you want to know where rare disease is happening within the Office of New Drugs, a lot of it happens right here, particularly in terms of genetic metabolic diseases. But we are certainly not the only place in OND where rare disease happens, because rare disease, of course, includes every possible specialty, from dermatology to oncology to pulmonology. And I would particularly highlight neuroscience that does a lot of rare disease neurodegenerative work. So there really is rare disease work that happens throughout the Office of New Drugs. Again, to break down DRDMG, we're really two parts. The Inborn Errors Review Group, where we have the medical officers that actually are reviewing the drugs and looking at the applications themselves. They're looking at the individual INDs, NDAs, and BLAs. And then we have the Rare Diseases team, where I work as the Associate Director for Rare Disease, where we're doing things like consult services that I'm going to touch on briefly, a lot of outreach and education as well. So first, briefly, I want to touch on, we're all here to talk about, or at least I'm here to focus on rare disease. So I want to talk about some recent approvals to sort of bring this down to the individual level of what's happening and what's happened in recent months with rare disease, because we are approving rare disease drugs all the time. So first, just to talk about tividenofusp alpha. This is a glycosaminoglycan enzyme, and it's used for the treatment of Hunter syndrome or mucopolysaccharidosis 2 in pre-symptomatic or symptomatic pediatric patients. This was approved under accelerated approval based on the reduction of cerebrospinal fluid heparan sulfate, which is believed to be the problematic chemical in this particular disorder. And as you can see on this slide, there was a single-arm open-label trial, 47 pediatric patients with Hunter syndrome, and it resulted in a significant reduction in the cerebrospinal fluid heparan sulfate. I think one of the things I want to highlight is we're often accused of, we only want to do double-blind placebo-controlled trials. I think when that's feasible, that's great. That's the gold standard. But some of these diseases are so rare that that's just not a feasible method to approach. So I want to highlight not only some examples, but later we're going to talk about things like the plausible mechanism framework, where maybe a randomized control trial is simply not feasible. But in this particular case, there were 44 patients who had measurements at week 24. Mean reduction from baseline was 91%. 93% of the treated patients had CSF HS levels, which were below the upper limit of normal. So a significant, and we thought clinically significant and important effect. Next, just to touch on pegzolarginase, this is an arginine-specific enzyme used in the treatment of a urea cycle disorder, which is called arginase 1 deficiency. This was another drug approved under accelerated approval based on the reduction of plasma arginine. There was a 24-week trial with 32 patients. So again, small numbers of patients, because just inherently in rare disease, we're often dealing with a very small N. The treatment with peglogenase resulted, again, in a significant mean reduction of plasma arginine, with 90% of the peglogenase treated patients achieving target levels below the upper limit of normal. And that was compared to 0% of the placebo-treated patients. And finally, as an example, copper histamine. This is a copper replacement product used in the treatment of Menkes disease. There were 2 trials. They were both open-label, single-arm trials, and they were compared to data from an untreated contemporaneous external cohort. The patients treated with copper histamine in the early treatment cohort had a significant improvement in overall survival compared to those in the external control early treatment cohort, with a 78% reduction of death. So just 3 recent examples that have transpired in recent months. And now I want to look at the big picture or long-term picture. If we look at approvals between 2015 and 2025, you see that almost exactly half of the novel drug rare disease approvals at CDER have been for rare disease. So lots and lots of development in rare disease going on. So that's the good part of the picture. I always try to balance this with the other part of the picture, which is that we have 30 million Americans living with a rare disease, roughly 1 in 10 people, and the vast majority do not have approved treatments. So while we've approved a lot, 700 unique drugs, 1,200 rare disease indications since 1983 and the passage of the Orphan Drug Act, still a long way to go and a lot of unmet medical need, which is something I'm going to repeat throughout this presentation. This is another way of just showing that same data. The bars in green are, or the bottom half of the bar, are the orphan non-molecular entities, new molecular entities that have been approved. The blue or the top bar are the non-orphan. And what I would draw your attention to is the purple line showing the percent of new molecular entity approvals that are orphan. And you see that since 2020, 50% or greater every year have been orphan drugs, so rare disease drug approvals. So again, a lot of approvals going on. But when you consider how many thousands of rare diseases there are, depending on how you count, perhaps more than 10,000, there's a long way to go. I think this is important, especially at an industry conference looking at expedited programs. So FDA has a number of expedited programs, 4 of them: priority review, fast track, accelerated approval, and breakthrough. And you can see that if you look at the left-hand bars or the red bars, which are the orphan NMEs, there's a higher percent for each of those of the orphan NMEs taking advantage of these programs compared to the non-orphan NMEs. And if you look at the right-hand side of the graph, you can see that 86% have taken advantage of at least one program and 69% have taken advantage of multiple programs. So I think what this says is that hopefully we've promoted the use of these expedited programs well. Industry has taken advantage of this. And maybe this is resulting in more and faster rare disease drug approvals, because I think that's the common goal that everyone has. Anybody familiar with rare disease knows that it's a very challenging field. Just to highlight some particular things, obviously we're always dealing with small populations by definition, but we're also dealing with a number of other challenges. The natural history is often poorly understood, so frequently we don't know how large populations of patients behave over time, simply because no one's ever studied that, and it's very hard to put together a large population if you have a disease where there's perhaps 50 people in the entire world that have a particular disorder. These diseases are often quite terrible, serious, life-limiting, and often progressive, just meaning there's a tremendous amount of urgent medical need. Many have pediatric onset. There's phenotypic and genotypic diversity, so not everyone has the same genetic mutation that has the same disease, and not everybody presents with the same signs and symptoms. So this makes studying it just that much more challenging. I mean, it'd be great if everybody with the same disease showed up at the exact same time with the exact same symptoms. That would make studying rare disease so much easier, but that just never happens. All of these diseases tend to be very genotypically and phenotypically diverse. Often there's not good translational background or animal models for us to study, and drug development tools, things like outcome measures and biomarkers, are often lacking. And since some of these things are very slowly progressive, it would be wonderful if we had a biomarker we could measure. That would certainly speed up drug development, but that's often just not something that we know about at this time, that we just don't have good biomarkers in many of these diseases. And then lack of clinically meaningful endpoints. Obviously, if you want a drug approved, you have to measure that the drug does something, and you need an endpoint to measure. And often what that endpoint should be or how to determine that that endpoint is something that is clinically important can be very challenging. I think the bottom line here is that rare disease drug development is very complicated. And add to this the environment of rare disease drug development, where we often are dealing with many newer and smaller companies that may not have experience working with FDA. That certainly can present its own challenges. On the plus side, we tend to have active patient stakeholder groups who are very willing to participate in trials, very interested in helping promote rare disease drug development. I think that's a huge positive that we should recognize. We should also recognize that there's often a dedicated academic community that's looking to promote rare disease drug development, but they often have limited regulatory knowledge, so that can also present another challenge. But the bottom line is I think we all have to work together because we all have the same goal, right, which is approval of rare disease drugs that are safe and effective. To that end, we've developed CDER's Accelerating Rare Disease Cures, or ARC program, with the vision of speeding and increasing the development of safe and effective treatment options, addressing unmet medical needs of patients with rare diseases. So at this point, I want to talk a little bit about the things that the ARC program, what it is and what it does. It really spans 3 offices at the Center for Drugs: the Office of New Drugs, the Office of the Center Director, and the Office of Translational Sciences. By bringing those together, really bringing together incredible institutional experience and brain power across the center. My group is that little dark semicircle at the bottom, the rare diseases team. I like to think of us as the connective tissue that holds those other offices together and spans across those silos. And another point I want to make, FDA sort of often accused of working in silos. This is exactly the opposite of what we try to do. Try to bring those offices together and work across offices to promote rare disease drug development. We also have the other CDER offices listed here and other FDA programs like the Center for Biologics and the Rare Disease Innovation Hub. We're working together with those groups extremely frequently and meeting with them continually to, again, break down silos and work across different offices and even different centers. The ARC program has worked hard to develop some resources, so I want to share some of those things. First is the ARC website. The FDA website is extremely comprehensive and very helpful, but it is also just expansive and not easy to navigate, or at least I don't find that it's easy to find what I'm always looking for. So we've tried to make the ARC website one-stop shop for rare disease information. As an example, we put together rare disease news and events. So if we have a workshop, as we just had a workshop this Monday, which I'll touch on in a future slide, we will post the recording there and the slides there as well, so that if you miss out on a particular event, you can go back and reference those. We also have a guidance page. FDA does have a guidance search page, but if you don't know the title of the guidance you're looking for, a particular keyword, it's very hard to find what you're looking for. So what we've tried to do here is organize it by categories. So those that fall into rare disease or digital health or clinical trials, so that people can find it. And we have a QR code which will take you right to the page, just to try to make that as convenient and as easy as possible. And we keep this up to date as new guidances come out. We add the relevant guidances to this page. I just want to highlight a few more recent guidances. I touched earlier on the expedited programs, so we have a couple of guidances focusing on accelerated approval and for determining whether a confirmatory trial is underway. I think if you're looking to see how to approach an issue with FDA or what FDA's thinking is, read the guidance. And I know that's sort of tried and true and perhaps even seems trivial advice, but given the sponsor submissions that we see, reading the guidance is good advice. And if you have a question about something that's in a guidance, that's something to address with your review division. Next, I want to highlight the use of the Bayesian methodology guidance. Again, often we're sort of accused of just being tied into double-blind, randomized, placebo-controlled trials. I think we're open to innovative trials. We're open to Bayesian design. If that's something that might be applicable to your rare disease drug development program, talk to the review division. again, consult the guidance. And finally, perhaps the longest titled guidance in history, consideration for the use of plausible mechanism framework in the development of individualized therapies that target specific genetic conditions with known biological cause. So what we'll call the plausible mechanism guidance. This is brand new and just came out in February, and I think is a really important and significant development in terms of rare disease. So I want to take a few minutes just to walk through some of the aspects of that guidance. Really, it just talks about how one would establish substantial evidence of effectiveness and evidence of safety, which are required for approval, based on what we're calling the plausible mechanism framework. And the guidance touches on things like non-clinical and clinical aspects of a drug development program. It also is very careful about which programs would apply. This would apply to those with a specific pathophysiologic abnormality serving as the root cause of the disease, in which is severely debilitating or life-threatening in a small number of patients where a randomized controlled trial typically is not feasible. So I think this doesn't apply to, say, your more common, quote, rare diseases, but is going to apply to those where you simply have such a small population that it's not feasible to do a randomized controlled trial. So 5 aspects to the application. You have to have a specific genetic, cellular, or molecular abnormality with a clear connection between that alteration and the disease indication. So some understanding of the molecular mechanism of the disease, not always available, but necessary for this framework. Then developing a therapy that targets the underlying or proximate cause, relying on well-characterized natural history of the disease in the untreated population. And I think this points out that if you're looking at a rare disease and thinking about the plausible mechanism framework, beginning to look at your population and document natural history, if that's not well documented, doing that at the earliest possible time point is crucial. Confirming that the target was successfully drugged or edited, and then demonstrating improvement in clinical outcome or course. So I think we look at each of these individually and very carefully, taking into consideration things like the clinical context for the disease, the level of unmet medical need, as I've mentioned several times, and the challenges involving enrolling participants in a clinical investigation when you're dealing with an individualized therapy. Again, typically dealing with very small populations. Approval is really based on the existing regulatory pathways, so that's why we often describe this as a framework. So you still have to demonstrate substantial evidence of effectiveness. Typically, that would be a single adequate and well-controlled study with confirmatory evidence. Depending on the endpoints there, that could use either the accelerated or traditional approval pathways. Again, one adequate and well-controlled trial with confirmatory evidence. Confirmatory evidence can include things like mechanistic or pharmacodynamic data, confirmation that the target was engaged based on clinical or non-clinical data, and exposure response data. Said this several times already, but again, this is expected to be used in a situation where there's a very small number of patients treated. We know that the primary source of the data may come from first-in-human trials, but that still means it has to be designed to be adequate and well-controlled by our regulations. And as I mentioned, I think on the last slide, early planning is essential, and you want to leverage all the data that's available. So initiating an observational study as soon as you identify study participants is key, and identifying any available sources of natural history is also very important, because natural history, again, is not often not well documented in rare diseases. The guidance then goes on to talk about some important considerations, things like establishing the genetic diagnosis in your patient population, trial outcomes and benefit risk, and also touches on the importance of data sharing, which I think is key when possible, because when you're dealing with such small patient populations, any data generated is really very important data. Now I'm going to talk about a different thing. The rare disease evidence principles came out around the same time as plausible mechanism. So I just sort of want to take a couple slides to talk about RDEP and distinguish the two. So the goal here is to facilitate approval of drugs for rare diseases with small patient populations and significant unmet need that have a known genetic defect as the major driver. There are a number of eligibility criteria here. So I would emphasize that you can apply for RDEP, but it has to be prior to the launch of the pivotal trial. So that's a key aspect here. And it also has to include these 4 things where the drug is for a very small rare disease population or subpopulation. It's intended to treat a known inborn genetic defect that is the major driver of the disease. The clinical course is progressive deterioration in function leading to rapid or significant disability or death. And there are no adequate alternative therapies. Substantial evidence effectiveness, as in most cases with rare disease, is going to be based on one adequate and well-controlled study. But we've stated, distinctly that this can be a single-arm trial together with strong confirmatory evidence of the drug's treatment effect. And again, confirmatory evidence in this case could include things like the drug's treatment effect on the direct pathophysiology, evidence from a relevant non-clinical model, relevant pharmacodynamic data, or even things like case reports and data from an expanded access program. So to apply for RDEP, you would submit a request to the existing IND, and a request for the participation in RDEP should be accompanied by a formal meeting request, and then accepted sponsors will have a meeting with FDA with the appropriate review team to determine what data can be used to establish substantial evidence of effectiveness. I'm now going to jump back from guidances to the ARC program and talk about our educational and outreach program known as Learning and Education to Advance and Empower Rare Disease Drug Developers, because we love acronyms, also known as LEADER-3D. So what we did for LEADER-3D is actually went out and talked to rare disease drug developers and said, where are there gaps about understanding what FDA does or about rare disease drug development generally? And then now we're developing educational materials to address those gaps. That information is documented in a public report, which is on our website. Really came out to about 6 things in that report. Things like natural history studies, things like clinical trial design and analysis, as well as endpoint and biomarker development. To that end, we've developed case studies. There are 6 published case studies on the website that again cover things like an example of a single adequate and well-controlled trial with confirmatory evidence, or leveraging external control. So these are examples of successful rare disease drug development that have reached approval. We've also developed 3 short educational videos. So these are like 5-7 minutes long. One looking at clinical study design considerations for clinical trials in rare disease, one looking at the importance of endpoints, and a third one looking at the use of natural history data. So you can find all those on the LEADER-3D website. I'll let you know that we have more materials in development, and we'll be adding those to the website as they become available. Another program that we are involved in is the Rare Disease Endpoint Advancement Program, or the RDEA pilot program. This is a joint program between the Center for Biologics and the Center for Drugs. And the idea here is to advance novel endpoints in rare disease drug development. Really 2 requirements. One is to have an active IND or pre-IND to address a rare disease. And the second is that you have a novel efficacy endpoint that you're interested in developing. So for each proposal that gets admitted into the RDEA program, you get an initial meeting and up to 3 additional meetings with FDA to develop your endpoint. And applications are accepted quarterly. So the current deadline would be June 30th. So again, deadlines March, June, September, and December. This started in 2023. The pilot runs through 2027. And we accept a limited number of proposals, roughly 1 per quarter or up to 3 per year. We've had about 20 applications to the RDEA program. 6 have been accepted. So if you're looking to develop a novel endpoint in a rare disease, this is a good way to get additional meetings with FDA to work alongside FDA and how to develop that. I mentioned earlier that we had a public meeting this week for RDEA, the Advancing Novel Surrogate Endpoints for Rare Disease Drug Development workshop. So I hope some people caught that, but if you didn't, that's okay. As soon as the recording's available, we're going to put it on our website. So you'll be able to tune into that if you have an interest in things like biomarkers or digital health or harnessing sources of patient-level data. probably worth your time to tune into that. I want to touch on two internal consult teams that we have, just to point out that we are working not only to do external education and outreach, but also working internally and across offices to look at rare disease drug development programs in a lot of depth and try to consider all the evidence that is submitted to us. So the first is the translational science team. It's composed of subject matter experts and leadership from multiple CDER offices. And really, we work with the review teams to try to do a very detailed evaluation of all the published literature in a particular disease and for proposed surrogate endpoints. Typically, we're looking at a biomarker here when someone comes in and is interested in accelerated approval, and we're looking at the evidence that supports that biomarker. And our goal here is to provide clear and pragmatic advice, both to the review team and to the sponsors regarding whatever the question might be. But again, typically it's a biomarker and accelerated approval. The second group is the Rare Disease Drug Development Design Working Group, or RD4. This one's really a pilot, so not really as far as along as the TST, but is a pilot consult service where we work with our biostatisticians and look for expert advice in the design and analysis of rare disease clinical trials. I think nothing is more sad than when we get a trial submitted and the results turn out to be uninterpretable. So the real goal here is to identify ways to improve trial design or do the trial in the best way possible so that we get interpretable results. If you're looking to find more information on any of these things I've touched on, I would refer you to the ARC annual reports on our website. We just put out the year 3 report in December and details many of these things that I've touched upon. And finally, I want to talk about the Rare Disease Innovation Hub, which is a relatively new entity at FDA. This is run by Amy Comstock-Rick. Amy couldn't be here today, so I'm presenting some slides for Amy to talk about the Rare Disease Innovation Hub. The idea here was to create a focal point to enhance synergy between the 3 medical product centers, CBER, CDER, and CDER-H, to strengthen collaborations across FDA. This is done through a steering committee composed of center director leadership and the rare disease leads from each of those 3 medical product centers. So we meet and discuss relevant issues. And one way we do this is looking at things like when CBER and CDER have applications for the same rare disease. We talk about how each center is approaching that, and is there a common ground, or because perhaps a gene therapy and a small molecule are very different, are we looking at those things in a different way? So we discuss those, similarities and differences, if you will, through the Rare Disease Innovation Hub. The hub really has 3 goals. First, the externally facing goal of advancing science through engagement. They've done this through RISE workshops, which I'll touch on at the next slide. Goal 2 is really internally facing, again, to promote cross-center information sharing and alignment that I just touched on. And goal 3, serving as a centralized point of contact for external partners. One of the criticisms have been that there's a multitude of ways to contact FDA about rare diseases. So the hub serves as a central point of contact. If you don't know exactly who you're looking for or who to turn to, they will triage those requests. So touching on their external goals, the RISE workshops, they've held 3 so far with a 4th plan for September 29th with the topic TBD. The most recent one was on data sharing. So I think these have been very interesting and informative topics where we're exchanging ideas. And again, this brings together patient advocacy groups, different aspects of FDA, as well as industry. And then internally, things like the Rare Disease Policy and Portfolio Council, which is where, as I was mentioning, CBER, CDER-H, and CDER all meet and discuss different applications that may have been submitted to us that might have some common indications. Here's some contact information for the hub. I would draw your attention, they do have a docket. So if you have a suggestion for a potential future RISE workshop, or you just want to make a comment to the hub generally, you can use that docket. And finally, I just want to leave you with some takeaway, 3 points. It's late in the afternoon. Sometimes gets a little sleepy, I've droned on for now 29 minutes. So in recent years, over 50% of CBER's novel drug approvals have been for rare disease. So a lot of rare disease drug development going on, a lot of drug approvals going on. The CDER-ARC program continues to work effectively with our rare disease drug development partners to accelerate effective therapeutic development. And we have multiple resources to assist with the challenges of rare disease drug development. And with that, I'd be happy to take any questions. Thank you.
All right, thank you for that presentation, Dr. Winiecki. At this time, if there are any questions in the room, please line up with the microphones. It does look like we have a question. So go ahead, and we're ready when you are.
Hello, Dr. Winiecki. Thank you for the great presentation. I know you mentioned at the end there the Rare Disease Innovation Hub. What is the stewardship that you plan to do with the CDRH? Because I know you kind of talked about CBER and CDER talking about different.
Yeah.
But I actually worked on pegzil-arginase, and we had to talk with the CDRH.
Exactly.
So
I think you've sort of answered the question, right? Many times there are things like companion diagnostics or devices that are used in conjunction with the development of a drug or biologic. So initially we hadn't incorporated CDRH, and we found ourselves reaching out to them and asking them questions. So now they've just been brought in as a full partner, which I think makes complete sense. So while I don't want to speak for the Rare Disease Innovation Hub, because again, not where I work, but I think it makes total sense to bring them in because so often with these new drugs that are being developed, there is some companion diagnostic, there is some device, and that's where CDRH plays a role. So I think it's really important to have them at the table.
Yes, thank you for that response. Again, any other questions in person? Please line up at the microphones this time. Any questions? Okay, I see one here in the back.
Thank you for the great talk. I guess my question may or may not be out of scope, but when it comes to rare disease research and approving clinical trial for especially trials with very, very little participants, I'm thinking of one disease with less than 30 diagnosed cases across the world. Do you take into account the ethical considerations when you're reviewing the drug? For example, there's nothing else on the market to assist this participant. Do you look at ethical considerations of whether the drug is going to be effective or it could be potentially, do you know what I mean? Any kind of potential conflicts of interest when it comes to like patient foundations who are also enrolling in the study, if that makes sense.
I mean, is the question about the feasibility of doing something that's a placebo-controlled trial versus like a single arm trial? Is that sort of what you're?
Something about like the ethics of like whether or not we can accurately assess the effectiveness of something if it comes to like a very rare disease with very little participants who are motivated.
Right.
For.
I think that also depends on things like the effect size of the drug, right? You know, in an example where you have something that's universally fatal in a year or two, and then you do a trial and you show that people are living to age 4 or 5 or beyond, or you have children who never did anything developmentally or even roll over, and now those children are walking. You don't need a huge trial to demonstrate that something has happened there, right? So I think it depends on many considerations, and you know, effect size being one of them. But I think, yes, we think about those things all the time. Really, I think we sit down, we're trying to objectively look at the evidence, but I think we're looking at that evidence completely in the context of that disease. And that includes how frequent it is, how it presents, the genotypic and phenotypic diversity, as I mentioned, the effect size of the drug. All those things are part of that consideration, absolutely.
Thank you. Sorry for the confusing question.
No, not at all. Thank you.
No, thank you for that. And again, we encourage any in-person participation. So at this time, please line up at the microphones. While you do so, if you'd like to ask a question, we'll go ahead and switch over to an online question at this time.
Can you please explain the differences between the ARC program and the Rare Diseases Innovation Hub?
Sure. And I think this has been a source of confusion because of similar names and because we're both focused on rare disease. The Accelerating Rare Disease Cures Program, where I work, is located within CDER, so it's a CDER program, and we're really focused on those consult services that I mentioned, outreach and education like the Leader 3D program, but we're all of our activities really focus around drug review, such as, again, doing those case studies, and talking about the details of drug review. So we're very part of the Center for Drug Evaluation and Research, very focused on drug review. The Rare Disease Innovation Hub really spans between those three centers, CDRH, CBER, and CDER. And again, I think they're looking at things like being a universal point of contact for people external to FDA who may not know exactly who they want to contact, as well as conducting those educational workshops and bringing the centers together. So the hub is really involved mostly, I think, on cross-center activities as opposed to within a single center.
Excellent. Thank you for that response. Another question in-house. Looks like we do have one here.
Thanks, Dr. Winiecki. So my question is on whether or not you think with the increasing trend of rare disease drugs getting approved, you have any thoughts and commentary on the evolution of FDA's regulatory purview on accepting more flexibility in their decision making. We're hearing things like single trial defaults, and you mentioned the plausible mechanism pathway, but extending that beyond maybe rare diseases. And does your office also do consults outside of rare disease?
Yeah, we don't. I'm focused exclusively on rare disease, so I don't touch on things that aren't rare. But I think you make a good point about flexibility, which is something I didn't really touch on directly. I didn't use the word flexibility. But I think we're always looking for how can we look at the data and fit that into our regulatory framework. So, right, part of RDEP is just saying we're going to use all available data and consider everything that's available to us, maybe even something like an expanded access program data in the evaluation of a drug application. So I think we're always keeping flexibility in mind while trying to maintain that strict approval standard that's outlined in our regulations, right? The standards to approve a rare drug are not different than the standards to approve a drug for lowering cholesterol. Those regulations are the same, but the way we look at the data, the context that we consider it in may be different. But I think when we're dealing with rare disease, we're almost always looking at one adequate and well-controlled trial. I mean, it would be not a typical situation to have more than one trial simply because of the rarity of most of the diseases that we're dealing with.
Thank you. All right, another invitation for in-house questions. As I don't see anybody at the mics right now, we can go over to another online question.
What are the benefits of participating in an RDEA program?
So the Rare Disease Endpoint Advancement Program, RDEA. So I think this is a really excellent program in terms of it gives you the opportunity to meet with FDA multiple times to develop your endpoint. My experience is that endpoints are one of the things that really can be a very challenging aspect to rare disease drug development programs, and having a good endpoint that hopefully you have agreement between a sponsor and FDA on what to measure and how to measure and what includes a clinically important difference. Hopefully you can establish all of those things, which really helps a drug program move forward. The advantage of RDEA is you get the initial meeting, as I mentioned, and up to 3 additional meetings to develop your endpoint. So I think sitting down with FDA and looking at your endpoint is probably quite a valuable thing. It certainly helps us understand what a sponsor is looking at, helps us understand how it's being measured, and the maybe advantages or disadvantages of that particular endpoint. So I think anytime you have the opportunity to meet with FDA, that's probably a positive thing, and RDEA offers you up to four meetings to discuss your endpoint.
Excellent, thank you for that. So this is our final call for in-person questions. If there are any in-person questions, if not, I think we have time for one more online question.
Sure. Do you think the rare disease drug approvals will continue to account for half of all novel CDER approvals?
I think that's a hard call because obviously that's dependent on the drug applications that roll in a particular year. But certainly the trend has been over the last 6 years that roughly half every year have been for rare disease drug approvals. I think there's every reason to think that rare disease drug development is going to continue to progress. Just living in the technological world that we do, right? Patients are able to find each other. Patients are able to come together. Whereas diseases that were so rare that patients never even knew there was someone else on earth that had their disease, Now they're forming advocacy groups, and they're funding research. So I think simply because of that, hard to know in any particular year from year to year, but I think if you look at the long-term trend, it's likely to stay the same or increase would be my guess. But that's just, that's my speculation. But my reasons for it, I think, are pretty solid.
Excellent. I think we can go ahead and take the final question in room. Do you
I
see
just
one?
had a quick follow-up to the other question that was mentioned on the RDA program. Does the RDA program accept digital or like devices inside their considerations for endpoint?
Oh, I'm going to have to look that one up. But I don't think there's any, as far as I know, there's no limitation on what that endpoint can be or how it can be measured. But I can give you the RDA contact person that you can reach out to. So I can give you her email and you can reach out directly with that question. But I don't think there's any limitation on what the endpoint can be or how it's measured, as long as it's for a rare disease and as long as that endpoint's novel.
All right. Well, excellent. Well, thank you for that response, and another round of applause for Dr. Winiecki.
Thank you for staying late in the day.
Of course. It's an excellent presentation to end on. So with that being said, we have reached the conclusion of REdI 2026. I do want to say thank you to everybody who attended here in person today, as well as everybody online. I know our room has been full on Adobe all day over both 2 days, so that's fantastic news. Also, people on the live stream as well. We've been paying attention to those numbers, so thank you so much for showing up. I hope you all learned something and took away something from this conference. As a few reminders before we close out, the live stream will be posted to the event webpage as soon as tomorrow. So you'll be able to see those recordings then. So keep an eye out for then. Also, the official recordings will then be posted to that same event page once they are available. Next, our certificates of attendance to support SOCRA, RAPS, SQA, and ACRP go out with the thank you email that would be coming out tomorrow. If you did a walk-in today and you had to write down your name, also make sure that we have your email address as well before you leave today. So you can stop by the registration desk to write that down. And that evaluation and certificate are available for 2 weeks. You have one chance to download it when you complete it, so make sure you do it right then. And then with that being said, that concludes today's event. Thank you again, everybody.